So, we will start with current processing and valorization strategy. So, before we go into this aspect, so we will go quick recap. So, are we all clear what is a bioeconomy? Yeah. All the economic activities to date, whatever we have been carrying out, so we have to have sources from biological origin, as simple as that. And circular economy, so whatever economic practices are ongoing, how we can make it circular. So, it includes everything existing processes. So, blue circular economy or blue circular bioeconomy. So, when we deal with the blue circular economy, most of the food operations will be automatically bio, whereas all other operations which are not of biological origin will be blue circular economy. Yeah. So, these three are important aspect. So, why do we need a bioeconomy? First, for energy, plastic, textile, chemicals and food. The issue is non-renewable, unsustainable resources. Because of that, we are looking for biological origin, that is relying mainly on agriculture, forestry, seas and valorizing waste and side stream. Yeah. So, that is new way of looking into the bioeconomy. Now, coming to this particular lecture, we are going to focus on current production practices for blue bioresources. When I say blue bioresources, what we are going to describe are different types of biomasses which are there for valorization, what all exist and how we process it and how we can valorize them. So, before going into this, so what do you think, what are blue resources? Can you give some examples? Easy one I can say, fish is one, shells or shellfish, yeah. Yeah, okay. Algae, that is both micro and macro. Any other? Somebody said the other day corals, yeah. So, there are number of species, some of the species even we are not aware of. So, how do we classify them? Because if you are from any of the stream, whether it's botany, zoology, chemistry, physics, classification becomes very important in order to say it. Yeah. So, majority of them are known as fin fish. So, which contain fins, that is small pelagic, that is herring, sardines, anchovies, medium pelagic, mahi, bonito. There are a lot of different species. There are only few examples, and that varies depending upon the water, depending upon the geographical location, and then large pelagic fish. You know what does pelagic word means? Pelagic is the area of the ocean which is not very deep and which is not very shallow. Yeah. Then you have salmonoids, that is salmon, trout, crab, tilapia, cod, which is deep water. For example, cod, haddock, pollock, demersal fish, that is deep in a water. And then we have crustaceans, we have cephalopods, mollusks, aquatic plant, algae, that is seaweed or macroalgae or microalgae, and other aquatic animals, and sea cucumber is a sort of seaweed. So, this is not the exhaustive list, but still there is a long list of species and animals and the plants which are present in ocean. And some of those species can also grow in fresh water, and majority of them are listed here are all for marine water. Now, if you will see all the species here, whatever we know about the fish, shells, algae, big fish, small fin fish, so all the data about them, what they contain, what for they are used, that is mainly coming in either from published or unpublished analytical data. Somebody like exploratory person goes into the water, brings some species, names them and do the chemical composition analysis, that type like what you guys do for any of those survey work, or nutrient value computed using a predicted model, because there is also seasonal variation, size of the fish, which area of the water species belongs to, or calculated values of food and species, in-house empirical measures using standardized method. So in case if we have to do the compositional analysis of any biomass, what are the key compositional analysis carried out? One is a moisture, which is having approximately 70 to 75% moisture is present in most all the species. Protein, 10 to 14%. Lipids, 2 to 10%, or it's not writing on the wall, it's not exactly this number, but it's broadly speaking the values are. And how much will be, what do you think, how much will be a fat content in shells? Almost negligible, they don't contain any fat, that's mainly a protein and other minerals. So again you have a ash content, that is mineral composition, again varies in a similar line as of lipids. So what type of carbohydrates are present? Do you think, do they contain any carbohydrate? Almost negligible. No, does animal contains any carbohydrate? Very small quantity of carbohydrates, mainly in the form of glycogen and other ones. So these are good sources of protein, lipids and different nutrients. So if you see the current production system, this is, I bet you won't like these type of statistics, same do I. Even I don't like the statistics, all these numbers, yeah. So if you'll see total world production, and it is almost increasing quite similar for inland, that is freshwater, and marine remains quite stagnant, the similar one. Same thing for aquaculture is increasing in both inland as well as marine one. That means rather than going for wild harvest, preferring to go for aquaculture, that is production into a localized location. So that means you are part of ocean, you start your aquaculture business there. So if you will see how much is intended for human consumption, that is an increase. Obviously we have to keep having higher amount of for food. Non-food application, per capita consumption is pretty much the same. X shares export and value for your sharing of export and import. So there are certain reasons they are overfishing. Can you guess which part of the world will have might be having overfishing? Asia is having huge amount of overfishing, including India. Is overfishing for certain species, and certain species are not used at all. So that is also a negative aspect of blue bioeconomy, that is we have to have a proper balance. So if you look at again with the production system, and if you see the ocean, and again this classification differs from who's explaining this. Geologists, geography will have different distribution, but as far as fish is concerned, most of the pelagic fish comes in this particular zone. Demersals will be all benthic, and all those different places will get fish. Majority of the fishing activities are within this zone, yeah, because after that there is again larger species and other species, large species, whale, blue whale and all those things appears. And some of the bacteria, microorganisms coming from the seashore also have a lot of pharmaceutical properties, lot of perfumes, lot of cosmetics are made out of it, and still things are getting explored. So it is not the end that we know this species and everything is done. That is done mainly on a terrestrial plants, wheat, cod, we know all about them, but not much about the aquatic bioresources. So it's still a huge open area for your research purposes. So if you'll see, this is a pelagic fish, and then there are different zones here. So majority of like epipelagic, meso, bathy, abyssal, hadal. So, accordingly you can classify. Yeah. So, pelagic fish is not a type of fish, but a fish which is grown in a specific area. Yeah. Something like if somebody is known as Indian, that means living in India, right? Or somebody living in Europe called European, but that European could be Indian again. Yeah. So, that is the way the classification are. But what is the actual classification for these fish? What will be the correct name? You know, we call it jhinga, machli, shellfish, and different names are there. So, what is the best identification is by using their zoological name, correct? Similar way, what we use it for botanical name for crops. Yeah. So, if you look at the marine resources, I will give you the example of Ireland, where I come from, which is surrounded by sea all around. The similar approach works for any country. I do not have the statistics and data for India at the moment. So, use of marine organism sources or the factory of useful, needful product and solution for societal challenges. This is OECD definition. Yeah. So, this includes your bacteria, fungi, other microorganisms, cyanobacteria, that is one way classified also as a microalgae, but this is basically a single cell bacteria, micro and macroalgae, sponges, mollusks, invertebrate, fish, fish co-product, and all other plants. Yeah. So, when you look into the literature, you might get something called sea vegetables. So, these are also a sort of macroalgae. So, if you go again back to back to the literature, and if you want to see how we can harness our ocean wealth, that policy document was prepared back in 10 years ago, and main focus was for thriving maritime economy, achieving healthy ecosystem. That means ocean, as we know, we did discuss yesterday, is a sink for most of the CO2s, and our engagement with the sea. So, any country is having a long coastline, even India's coastline is very long. Do you have any idea what will be the coastline of India? So, you can check that, even I do not know exactly what it is, but here in this case coastline is 7,711 kilometer for Ireland, and maritime territory, that is territory in and around a country, is considered as marine territory for a given country. That is water, you would have seen all those Indian water and international water. International water, that means it does not belong to anybody. The range is, I think, 200 kilometer or something, and that is guided by international protocols. So, with this you can see what is marine bioresources and how we process them, and this is really very difficult to identify what is exact processing method for each and every species. Every species is different, something like you are trying to study the food science in one lecture. Yeah. So, it becomes very difficult to cover each and every aspect how it is processed. So, we will try to do our best how we process them. So, first and foremost is harvesting. How do we harvest? So, fish one, I am not going to deal much into the fishing side of it, because majority of you are aware of it, whether you take a fishing net or you take a small what do you call that, having a small bait attached to anchor and throw it in the water to catch a single fish for recreational purposes or for food purposes. So, there are different types of techniques which are used depending upon the company or depending upon your capacity or depending upon the industry. So, it is individual farmers, they go with the smaller ships, or the big company which is having a commercial businesses, they may go with the big trawlers. So, this is the fishing part. When it comes to macroalgae, it is either wild harvest, you go to the beach, collect the plants, yeah, and they grow on the rock. This is called wild harvesting. As what we discussed yesterday, if you go in a forest, you see any fruit plant, you pluck fruits, that is wild harvesting. But if you grow those one into a sea, then it is called cultivated or aquaculture. How many of you have seen aquaculture farm in anywhere in India or some anywhere? Okay, they will share their experience. How does it look like?
There are different types of aquaculture systems. Some kind of species will be growing in stagnant water like ponds. Some will be growing in marine conditions, and some will be growing in running water like rivers. They for culturing the fish in a closed environment, we have to simulate that environment. Pond culture will be there. For that, a blue lining sheet will be a pit is digged, and a blue lining sheet will be
at
spread
the bottom
over that, and the water is filled, and the TDS and the pH of the water is maintained for the which is equal to the pond. Then in case of for simulating the river condition, the race culture will be there. A cement tank will be built, and in between a wall will be taken. The water will be flowing around the wall. The flowing water will simulate the river conditions for the animals. In case of marine conditions, we can occupy the area in the sea, or else we can add the salt. There will be some salt packets. We can add the packets to the pond culture in our area, or we can directly go to the sea. We can occupy the area of the sea, or we can simulate the salt conditions of the sea in our pond itself.
That's very good. So it's something like in Mutukadu, the people they grow, they are rearing crocodiles. Yeah? No? They're shells and.
In Mutukadu Siba, in Chennai, they are culturing these crustaceans.
Shells.
Yeah.
That's very
for
good.
Crabs and.
So you are well aware of aquaculture system, yeah? And there is actually also a guidelines how aquaculture systems are grown, because if you are using maritime or marine area, so when you are blocking the river water, wastewater coming in, it concentrates. And so those are the issues as well which has to be addressed for the production. So have you seen the seaweed farm, seaweed cultivation in aquaculture? So that's I'll be showing you some stuff how this particular seaweed is cultivated. If you will see this here again, how the seaweed cultivation is carried out into the sea. This is the image which is taken from using drones. Yeah. So if I'll just run a very short video, and I worked a lot with an industry, and then you can see how this seaweed is grown. So whether wild harvest, as you can see, this is that's me, and with the old Irish industry person, how he grows or wild aquaculture seaweed as well as wild harvest, and this is what is aquaculture. So as you can see, this is again a Dulra Marine is a company where a rope where the spores are added in, thrown into the water, and when it is fully grown, it looks something like this. So that means a single spore which germinates, and some of the wild harvested one which you see at the shore, but at the bottom of the seabed also you will see all these seaweeds, how they are grown at the bed of the seaweed. So what are their advantages of this seaweed? It's not only for food purposes or application, they are very, very important for protecting the shorelines, protecting your flooding or what you call tsunami, and all those flows are everything is stopped because these plants, and they grow rapidly, and they have one of the biggest advantage. When they grow, they do capture lot of CO2 from the environment, yeah, because they build a biomass. From where they get? We don't put any nutrients to them. Do we put any fertilizer? No. What is required is only the salt water, and rest all nutrients are covered from there. So as you can see from the seabed, so this is all how the seaweed looks like, and species could be Laminaria. No need to worry about these names. I'll cover this one when we go in depth. How do we produce these seaweeds and how it is done in IMTA system or in aquaculture. Laminaria saccharina latissima, you have Ascophyllum nodosum. So some of these species are And it's not that I went in to get this video, one of our colleagues went in to get it, that's why this is having all these seals and everything is found. So this is the way how it seabed looks like, and all these species are grown and they are consumed. So I am not going to carry forward this full video. Now this is again, this will give you another idea. This video is again from an American national agency. It talks about what is really a aquaculture. Originally aquaculture was breeding, rearing and harvesting of fish, shellfish, algae and other organisms in water environment. That's very simple definition. That is culture, that is cultivation in water bodies. Yeah. So this is very short video which shows how this aquaculture is carried out. Yeah. And now what you will see in this case, this is what one of your colleague was mentioning about cementing and making water bodies, raising, and this is sometimes the mesh side stuff is seen, and you grow fish or shells. Shells are recovered and this is the cooking process, and then this generates the waste, and when waste is generated, shells are separated and meat is separated. And if you go on to this NOAA, and you can find lot of different types of educational videos. Yeah. If you want to, if you're really interested how things are grown. Yeah. So it's all, and this is the best way to explain aquaculture, otherwise the design of aquaculture system and all again is a lengthy system, and I'm trying to cover most of the production in a shorter duration. Do you have any question related to this so far? So this is yeah, this is pretty straightforward anyway. So if you look at yesterday I mentioned about IMTA, that is integrated multi-tropic aquaculture system. So either aquaculture, as your friend mentioned here about the shells or seaweed or for fin fish, that is integrated multi-tropic aquaculture system. This is from this particular paper, this is schematic diagram, and details of this are from C Bioplus. That is, this is a project which was working, there are multiple number of projects they are running across Europe and other international projects dealing with what is the best way of growing or producing aquaculture. And the next, this image is from Marine Institute. Like TEAGASC, we have Marine Institute, that's Forest Namara. Mara means Irish word for marine, and institute. So that's Marine Institute, and website is marine.ie. So they are responsible for all the maritime activities and everything related to that. So this shows the clear picture how the IMTA system works. You have your fish, sea urchins, lobster, sea cucumber, kelp. Kelp is another name of seaweed. You would have seen green seaweed, red seaweed, brown seaweed. So these are all the color coding of seaweed. Yeah. And then scallops, oysters. So all these things can be grown together. That makes it IMTA system. That is integrated multi-tropic aquaculture system. And within that there are 5 or 6 different ways of doing aquaculture. And Japan, China and Southeast Asia, they are very well advanced when it comes to aquaculture. Yeah. So this is all how these biomasses are produced by aquaculture. So now this is just a glimpse of seaweed aquaculture process. First one, if you will see this particular rope, you have spore inoculation and seeding is carried out on a rope, and then this rope is thrown into ocean. I just leave it there, and after 3 or 4 months, what you do, all this hold fast, that is again the way it holds the rock or a rope. Seaweed is grown, harvested, and then this is the way it is dried and then ready for use. So the rope, spores on it, throw in the water, forget it, and then come back and collect it. So that's the way seaweed is processed. So we have one full lecture, I think lecture number 10, on dealing with seaweed production I'll go through all the steps of seaweed production as well as microalgae production, how it is being carried out in lecture number 10 is scheduled. So, it was only to give a glimpse how the seaweed is produced. So, what is seaweed used for? How many of you have consumed seaweed in any form or the other?
Dairy products, like we will be using.
That's ingredient like carrageenan and alginates and all those things are there, right? Any other? How many of you have eaten sushi? Yeah? The outer one is the seaweed, that's nori, yeah? So, that seaweed is used indirectly or directly, everybody consumes it in some form or the other. So, majority of the market is to do with the food and beverage, pharmaceuticals, animal feed, fertilizer, cosmetics and personal care products. So, as you can see, Asia Pacific is the biggest one for seaweed production, cultivation and processing. So, it's all, this shows the economic value and who is the leader in seaweed. Rest of the world is very teeny tiny 0.2%, whereas China is a 62.8% and rest of the Asia is 37. So, that is why Europe was not using the seaweed apart from getting agar-agar for lab cultures. So, everybody has used that particular media. If you have done the experiments, agar-agar comes from where? From seaweed, yeah? Any idea which seaweed it comes from?
Gelidium and Grasslera.
Gelidium scavona, like Gelidium with S name is there, that is, you boil a seaweed at 110 degrees Celsius for 5-6 hours, whatever comes a liquid portion, you cool it down, it forms a gel, that becomes your agar-agar, yeah? Alginate production is slightly different, carrageenan production is quite similar to your agar-agar depending upon what seaweed we use, yeah? So, it is not a complicated process to extract those biomasses. So, any question related to seaweed production? Anyway, we are going to cover the full seaweed production, both macro and microalgae briefly when we go into the lecture number 10. So, just wanted to show the seaweed production is not, it is bit energy intensive, but it is not having any input. So, that means all the CO2 for environment is accumulated and it cultivates the biomass and you get carbon capture, that is carbon dioxide from environment is captured by sea plants and then it offsets the carbon to produce a biomass. So, that is one of the way of sinking the carbon by increasing aquaculture production, but it requires the license. So, every country, every place there is a different regulations. I do not know what is the current regulation here, but normally if your house is next to a sea, certain areas of the sea belongs to you. So, that was the policy. Think is same way if you are living in a forest, that forest area belongs to you. Similar way it was with the sea, and then if you have to start any aquaculture system, you need to have a license, because that is again impacting the environment, recreation activity. So, you cannot start your aquaculture system on Marina Beach where people are going there for recreational purposes, yeah. So, these are some of the limitations. So, now, if you will see about production of macroalgae, as I said in lecture 10, Bantry Marine Research Station, this is our industrial partner which produces macroalgae as well as small aquaculture seafoods, and that is located in south of Ireland and over here, which is almost 275 kilometers from Dublin, yeah. And this is the Bantry base. So, if you are interested in all their projects, you can go into their website BMRS, that is Bantry Marine Research Station, that is a privately owned company, and I have requested them to provide me some of the information of their production processes, which I have, which I will be running in lecture number 10. So, you can see in reality how it happens, not a theoretical one, yeah. theoretical one, you can easily look into the literature and you can have a look at all the production processes. And it's quite interesting to see how the seaweeds are grown. And that's their building, and that's the sea which belongs to them for production. Yeah. So this is for production of macroalgae. Now coming back to animal one. So out of you, how many of you consume fish? You can raise hand. 1, 2, 3, 4. Majority of them, yeah. And rest are vegetarian, I presume. Yeah. So cultivation of seaweed, microalgae looks nice, is green and all, and fish is extensively processed. You all know about the processing. So how fish is processed in India? How we process fish? Suppose somebody is harvesting a fish, it goes straight to your kitchen and you cook it, right? What is a commercial way of processing fish? See, it's not, I'm not checking your knowledge and all, because I am also trying to learn, because there is some other better method is there, we can adopt it. It's a two-way process, you know. Yeah. Go on, madam.
First thing is we have to maintain the cold chain, sir.
Okay, that's fine. You get a fish, how it is processed commercially? Yeah, cold chain and all is there, yeah?
Yes, sir. After harvesting, the fish will be frozen in the sea, and while it reaches the shore, after that we have to go for thawing process. And then after thawing, we have to, sometimes before freezing, they will remove the gut parts and the heads and the gills, all those things are removed. And in some cases, it is frozen as whole, and after thawing, the rest are removed in the processing plant. And after, different types of cuts are there, fillets, different types of cuts are there. And in case of shrimps, for that also different types of cuts are butterfly cut, tail on, like that, different cuts are there. After the cutting and further processing, they will be again frozen for further use. This is a kind of processing, and again we can go for pickling, like
salting, another one, or drying of fish, another one, right?
Yes, sir.
So this shows everywhere the process is different. Yeah.
Yes.
In majority of the European countries, fish fillets are sold in a frozen condition. Yeah. Raw fish is normal. Fisherman goes to the water, harvests the fish, sells the fish. You buy it, come home, you cook it whatever you want to make it, for curry or fish and chips or different product. So if you'll see different type of seafood, so she has already explained. So first one is to have keeping it in a cold condition. Yeah. If it is not in a cold condition, what happens? It starts smelling. It starts smelling because of TMA. Yeah. Trimethylamine. And this is difficult to remove. Yeah. And when we are freezing it, you freeze it, and when you are thawing it, you are thawing it for at least 7 to 8 hours for thawing process. So during thawing process also you get some of the microorganisms gets activated. Yeah. So there are one. So what are the processes how we can increase the shelf life of frozen fish? Either wash fish in chlorine, which is not allowed in most of the places. Yeah, even in Europe it is not allowed. So fish comes in, straightway you wash it, that is called stick water. The muddied water on the fish called stick water. Then you remove the tail or a head, that is deheader or detailing, just to remove the portion of unedible portion or remove the scales. Yeah. Then you fillet it, and that fillet is vacuum packed, goes for sale. Yeah. But in some cases when you are freezing the fish, the majority of the places the fish, the moment it is harvested, straight away it goes for processing. There is no storage in between. Or in some cases it goes straight for frozen condition. Then if you are using ozone in water for freezing, so during thawing process you will maintain its microbial stability, because during thawing process you need a normal temperature, and whatever microorganisms are not growing during the frozen condition will get activated when they get a conducive temperature. Yeah, that's the way you have to design a preservation strategies. Yeah, there is no pulse electric field, no high pressure can go in there, because if you take a fish, put in high pressure, you cannot make a fish cakes, because the protein is completely different. Yeah, whereas if you put seaweed into high pressure processing, how many of you heard about high pressure processing? Yeah, 400 to 600 megapascals condition. If you put a seaweed at 600 megapascals, even for 10 minutes, nothing will happen to seaweed. Whereas if you put a fish, meat or a carrot, it will become a paste in 1 minute. Yeah, because seaweeds, where they grow at the bottom of the sea, so they are structurally very, very robust. So they can withstand huge amount of pressure. Yeah, as what we were discussing yesterday, when you go into scuba diving, you start feeling the pressure the moment you go below 5 meters. So coming back to this stuff, so all the biomasses capturing, that is harvesting or aquaculture, and then industrial processing, packaging, and then distribution, wholesale, retail, consumption at household. That is pretty much very simple processing methods. Yeah. So what is applicable to fish may not be applicable to oysters or lobster. Yeah. Lobster or a prawn, they have a meat inside the shell. Is there any way, what could be the processing technique that we can remove the muscles very easily from those lobsters? You have seen the lobsters, right? Yeah. You have to break the shells or a crab to remove the meat. Yeah, and you have lot of muscles. So if the whole crab or whole lobster or this particular product goes into high pressure machine along with the shells, give a pressure, and then what you will see, if you remove shells, will come out like a shell of a peanut. It's just because when protein is, it expands, shells is robust structure, and then when pressure is released, it contracts, so it becomes easier, it detaches its muscles from the shells. Yeah, so that's the way it works. That is pretty much used in industry at the moment also to help in releasing the muscles. Yeah. So this was the easiest part for explaining the processing, because your colleague already mentioned. So main important thing here is preservation. And other element, especially for sardines and tuna, you will see in the market they are sold as tuna in sunflower oil, tuna in brine water. So fish is steamed, and that steamed fish goes into a salt solution and it is sold in the market. Yeah, that's canned fish or canned in oil, canned in brine solution. So number of different methods are there, and if you are in food product development and other ones, and you have your own way of doing so. Similarly, you have fish infused with other type of oil to make a different product. Yeah, so that is all product development, culinary application. The huge number of ways we can process seafoods. Now, so what happens when we have all these processing, whether it's seaweed, crustacean or fish bycatches, yeah, they all generate different types of product. Seaweed and current uses and process, as you can see, is agriculture, animal feed, fertilizer, sushi or salad, agriculture, biofertilizer, cosmetics, compost, soups. Yeah, there can be multiple number of products. Yeah, then there is different economic values related to that. These are all estimated based on how much products are prepared by using a minimal processing. Yeah, fertilizer is the most easiest one and important one to get a calcium rich fertilizer or so. Then how we can improve the economic value of current processes by valorizing the side streams produced by any of those biomasses. So yesterday we did discuss whatever bycatches we have, those bycatches can be converted either fertilizer or to get a protein or to get a nutrient and then can be valorized. Because of those issues, now wild harvest has been on reduction and aquaculture is on increase. Yeah, makes things very simple. Now, when it comes to seaweed, so you can focus on alginate, fucoidin, agar, bioplastic films. I'll cover some of those biorefinery aspect later on today or in the next few lectures. Then when it comes to crustaceans, you have chitosan, yeah, calcium carbonate. We'll do one of those experiments here, astaxanthin and collagen. What is collagen?
Muscle fibers.
How do we extract it? Difference between collagen and fish protein.
It's mostly present near to the skeletal muscle, attached to skeletal muscle.
Okay. I always go for the simple definition. Collagen is also a fish protein. Yeah?
Yes, sir.
Yeah, and this is all about in the shells, scales and all are there. And this is actually collagen is also used as a fish gelatin, correct?
Yes, sir. Yes, sir.
So you have gelatin, which is again comes from either in market as pork gelatin, beef gelatin and fish gelatin. So fish gelatin replaces other two gelatins as far as products are concerned. Then when it comes to your fish, you have fatty acid, bioactive peptides and protein. Some of you guys are working on nutraceuticals and other products, yeah? So all those fatty acids and peptides. So what is a peptide?
It's a polymer of amino acid, like bipeptide bond. When multiple amino acid combine to, even two amino acid combine to form dipeptide, like that.
Okay. So what is a protein? Protein is a macromolecule which contains nitrogen. Yeah. So how do we estimate protein?
Method. Sir, by Kessel method, we first find the nitrogen content, then we multiply it by some constant, then
By
we get the protein.
Zeldal method, you measure nitrogen content.
Yes, sir.
And you multiply by a factor, correct?
Yes, sir.
Right. And how we measure a soil nitrogen? Same method. Yeah. Zeldal method is used to measure a nitrogen of food as well as of soil. Yeah. So either we use 6.25 factor, yeah, most commonly everybody uses that, or 5.7, 6.35, 6.38. How do you choose these factors?
Sir, some of them from plant nitrogen, based on plant and animals, sir. Plants have less than 5 and animals have more than 6 or 6.25, sir.
How? From where we get this factor?
So it's a conversion factor. So based on most probably there are different sources of nitrogens are there. So apart from protein, there are other sources are there. So if you are if other nitrogen sources are there, accordingly we will be calculating like.
Okay, that is correct. Basically, we get these factors based on if we have to do any estimation. This is the, you say, beauty or limitation of all analytical method. We estimate some nutrient and we relate it to a specific thing. Yeah. So all the factors for animal products are higher because they contain lot of non-protein nitrogen. Yeah. If 14% of nitrogen in a body comes from the protein, so factor becomes 100 upon 14. Yeah. If majority of the nitrogen is coming from 18% of nitrogen in a body is coming from nitrogen coming from protein, then 100 upon 18. Yeah. Yeah. So that is the protein nitrogen and non-protein nitrogen. Can you name any non-protein nitrogen?
Ammonia, urea.
Huh?
Ammonia, urea.
Yeah, that is all byproduct one. Yeah, correct. but if animal did not eat anything, so you will not find those one. So basically your DNA, deoxyribonucleic acid, RNA,
Ribonucleic acid.
ribonucleic acid. You know purines.
Purines
What
and
are they?
pyrimidines. Adenine and thymine.
Correct. AT and GC, yeah, and pyridines, yeah. All these are non-protein nitrogens, yeah. So because of that, we get these factors. So it's not, Zeldal is only a method to estimate nitrogen content. So what are other methods of estimating protein? Bovine serum albumin, yeah, Bradford, yeah, Lorry method, they all have a certain limitation. They only estimate a protein containing tryptophan or containing specific amino acids, and that's why all the methods they have limitation. So the best way is to get all the amino acid profile and then to see this one, right? So protein is basically, that was not the intention of my question, because my question was related to peptide. Protein is basically a polypeptide, correct? Something like a polysaccharide, yeah. So starch is a polysaccharide made up of glucose, yeah. So how many glucose will make it a polysaccharide? More than 10, yeah. Definition wise, you have monosaccharide, disaccharide, trisaccharide,
Oligosaccharide.
oligosaccharide 2 to 10, and then above 10 is polysaccharide. So same way it is a polypeptide. That's why the protein molecular weight becomes very, very important, yeah. Almost all the biological toxins are proteins. Do you agree with that? All the natural toxins are protein, which cannot be digested by human body, and they block the stuff. Venom is a protein, yeah. Spores are also made up of fully of protein. So the larger the protein, it blocks the system, causes toxins, yeah, right. So that's why the protein is broken down into peptides, and peptides, they have a biological activity, which is the way how we break the structure. So I'm not going to go into it, but if you extract the fish protein with a specific enzyme, you can create a peptides, and those peptides, they have a biological activities, yeah. So we can get all the proteins, lipids, peptide from fish side streams. You have seaweed stuff, and you have from shells, yeah. So astaxanthin is because of the pigments present in the shells. In shrimp, you will find that one, and this is soluble in non-polar solvent, correct? You cannot get it in water, and they are quite expensive ingredient for different applications. So this is the way how we have a current economic value, what we obtain from there, and how we can improve its economic value, yeah. So what other things we are doing in terms of circularity? We are valorizing side stream, so we are reducing the impact on the waste and creating new biomaterial, yeah. Now, so what is the best way of sustainable exploitation? The beauty of this diagram is you just change the biomass here, yeah. Whether it's aquaculture seaweed or any of the shellfish, yeah. The seaweed is added because wanted to show the CO2 sink. You convert that into different products, direct product application or different ingredients. You fractionate it, yeah, and then you can create multi-product biorefinery, that is for feed, food, pharmaceuticals or drugs, skincare products, additives, yeah. So if you take a seaweed, different application, you have and say extract alginate, leftover biomass is having fiber and a protein, can be used for feed purpose. If it is used for feed purpose, that particular feed for animal can be used for making a food. That's again closing the loop. If you remember, chicken feed, they contains, how many of you eat eggs? You don't eat eggs, yeah, okay. So eggs, in order to have a proper eggs, you need to have a good shells. Yeah, that means you need to have calcium rich diet that can come from shells. There are some of the companies who are producing calcium for chicken feed just from the shells. Yeah, so that particular goes into food and feed. Pharmaceutical, again in a form of peptides, astaxanthin, fucoidin is sulfated polysaccharide which contains lot of antiviral and lot of medicinal drugs are made out of it. Alginate is also used, something like aloe vera cream and hand creams and lotions. That is another one. Additives, carrageenan and all we already discussed. And whatever is left to our biomass of that can go for biomass production or anaerobic digestion. Yesterday your colleague was explaining, and all those greenhouse gas emissions goes as a CO2 sink for production of seaweed. Yeah, and study of all these input and output is carried out by using LCA. Have you heard of this term? Life cycle assessment. Yeah, so that means you make a boundary and see what is coming out, what is going out, and what is that impact in terms of greenhouse gas emissions and different priority. That's the way it happens. Now, when it comes to seaweed harvesting, whatever way, first processing, that is washing, milling, drying, and seaweed meal that can be sold for whatever purpose we want. Yeah. Then again processing, packaging, and retail. So all these processing element depends upon what we are using the ingredient for. Something like a wheat which is harvested, wheat is sold like a grain, A product, or it being sold as a flour, B product, and the flour is made into your bread, cake, biscuit, whatever, that's another different product. Yeah. So simple one is, critical part is how do you harvest it and how you do your primary processing, then you can divide the level of processing depending upon the product. So whatever product you are using, if you are using seaweed only for energy, it is having low value application. Yeah, I know energy is expensive, but burning something for electricity or for stuff is low value application. Similarly for fertilizer, feed and food, the moment it goes to nutraceutical, cosmeceuticals or high value chemical, you are adding value to that particular ingredient. So this is the way how we can make an economic value of product. Yeah. So if you will see this particular one, biotransformation, one element is primary processing, which includes mainly cleaning, grinding, washing, milling. Yeah. Can be again secondary processing, that means you are converting that into a specific ingredient, or tertiary, that is trying to get a targeted ingredient. So there is again no borderline what will be the final product of a seaweed. In this case the final product is dried seaweed or milled seaweed or a powdered seaweed, that is a ingredient itself. For secondary processing, from powdered ingredient to specific type of plant stimulant, animal feed or extracted compounds. Third one is, once you have that particular stream, then you can create number of different ingredient for different applications. Yeah. So once we have this, so where all we can use again for seaweed, this is again what we discussed yesterday, nutrition for sustainable and healthy diets, because they contain lot of valuable nutrients. And then you have climate smart and environmentally sustainable food systems, circular resource efficient food system, innovations and empowerment of communities. Yeah. So by doing so, we are able to make use of every fraction of seaweed, and similar type of protocol or approach can be made for different species. And if you go into the literature, you will see multiple types of review papers are written based on these biomasses. Now, if you see how the seaweed macro and micro algae are used by different companies, human food, if you see the diagram, it just shows how the relationship between microalgae and macroalgae. Hydrocolloids. What are hydrocolloids? The starch is also hydrocolloid. Yeah. Simple definition is any ingredient which makes a colloidal suspension when mixed with the water becomes hydrocolloids. Yeah. That's the hydrocolloids. And then cosmetics, food supplements, nutraceutical, feed, others and pharmaceuticals. So, both macro and microalgae are used for different applications depending upon the sector. Now, utilization. So, majority of the seaweed production is used for alginate. Alginate comes from where? Which species? I never mentioned the species, how do you know it? Alginates comes from Escophyllum, Saccharina latticima. Yeah, some of the seaweeds, they are very good source of alginate. Every seaweed will contain those type of polysaccharide, but some are known for large quantity. Carrageenan is again from Irish moss or Chondrus crispus, specific seaweed, and agar from Gelidium. And there are certain species which are grown even in watery sargassum and all, they also contain these type of species and use up to 15 to 20% of world seaweed production only for these additives which are used in different food product applications. And then if you will see in last 15 to 20 years, more than 2,600 new product has been introduced into the Europe, which wasn't the case. And as I mentioned yesterday, anything above before 2008 were not under novel food regulation. So, it was not a market for seaweed, but now people are drifting towards more sustainable food products and ready to pay premium. And 1 kg of seaweed at the moment costs nearly about 40 euros per kg. Is very expensive, more expensive than what you can get a wheat or a rice at the moment. So, demand is growing at a faster rate and dependent upon we are using for fertilizer or for pharma purpose. I will give you an example of an ingredient called fucoidin. We transferred one of the process to industry. This is known to have around 2 to 10% in fucus vesiculosis. 1 gram of this ingredient on sigma chemical costs nearly about 400 to 500 euros. Yeah. And now this particular ingredient is used in Brown Thomas, lot of hand creams and different cosmetic products. Yeah. So, this is the utilization part of seaweed, whether it is your red seaweed, brown seaweed or green seaweed. Can you give an example of green seaweed? But technically all will be looking green, yeah, but they are classified as red, brown or green. Ascargopsis is basically a reddish color, brownish one could be a Escophyllum and green one is UVL vulva. Vulva or Palmaria palmata, these are the species and they are used for protein, carbohydrate, phenols, pigments and different applications. Yeah. So, for each and every species and each and every process, there is a specific process line. It is very difficult to cover all the processing aspect, but the critical part is washing, drying, milling and the story for fish, harvesting, cleaning and then it goes straight for consumption. And that is one of the reason why losses at the consumer end for seafood is quite high, because we are using it and then we have byproduct which goes straight to bin. Yeah. Any questions so far? Okay. I'll just show you quickly a history of the seaweed product. Back in 1845 when there was great Irish famine, because majority of the diet in Ireland was based on potatoes. Like in north, you know, if you haven't eaten a chapati, you haven't had your food. In south, if you haven't eaten a rice, you didn't had a food. Same way in Ireland, if you haven't eaten a potato, then you didn't had your food, because potato was a staple crop. And when there was a big issue at that time, dulse, that was the seaweed that's called Irish moss. Clondus crispus is known for carrageenan, which was used as a food purpose. And now the commercial products, the lot of different types of commercial products are available as an additive or as a direct product application. Even this, you can see this particular chips and crisps were developed. And same way, this type of Alaria esculenta is a seaweed which was used directly into making a bread product or extruded snacks. Yeah. So these are all direct application of products. So this will be the last one for this lecture. So what are the bottlenecks and what are the opportunities for seaweed products? One, this is the problems of Europe. It's not pertinent to Asia. One is there is a huge competition for aquaculture production from Asia, because there's well advanced China and Southeast Asia, well advanced in aquaculture. Manual labor is used at the moment for aquaculture. That's a huge energy intensive because labor is very expensive. Complex or non-existence of national laws. Earlier, anybody whosoever is living near the sea, whole sea is theirs. They can use it. That's the way rules work. And then license, and there is also a issue with arsenic and iodine content. Yeah. The only advantage of European seaweed compared to Southeast Asian seaweed is presence of heavy metal is huge over here compared to what is there in northern hemisphere, because that area is not used for vehicle transport, because this is particular Southeast Asia is used for transportation purposes, yeah, for business. So that's another issue. And very few species are used for cultivation. Yeah. Others are present in water, but they are invasive species. That means the species they came in through the water, but they do not belong to native place. Since the new species comes in, that creates disturbances again into the ecosystem. And then how do you do the breeding program to have a robust seaweed structure? And issues how we can create a biorefinery for seaweeds. Yeah. Even we haven't developed a biorefinery for cereal crops, how we can develop it with this. So with this particular one, I'll close this particular lecture. So do you have any questions so far? So if you have any question, keep it in mind, we will run it when we go in afternoon. So what we covered was what are the bioresources, A, B, how we process it, yeah, processing and aquaculture, yeah, and what will be the best way to do sustainable exploitation of macroalgae. So when we come back in afternoon, we'll have one or two recap slide, recap work from biorefinery aspect of algae, and then we carry on with how do we identify the waste, how best we can valorize it. Yeah. And if you have any question, write it and keep it, and we can go through in next session. Thank you. So far whatever we were focusing on in the previous session yesterday was all about your bioeconomy, and this morning we were covering what is production and processing aspects. Yeah. So quickly, what is basically a processing aspect for fish? It's pretty much straightforward. Main focus is on harvesting, preserving at farm, yeah, that is cold water and refrigeration and freezing, washing, and then product is ready for consumption, that is again at the domestic level, yeah. And some of the commercial products are formed, and different products and different applications or different sources, that is source for animal feed, fertilizer, and what else was there? Animal feed, fertilizer plus food, yeah. And then whatever side streams are left over, they are used for making again high value ingredients. Many of you are doing topics somewhat related to that, yeah. So within within this we also saw the biorefinery for seaweeds. That means how those seaweeds can be used for different functionality, different application. So one thing which was was not covered was related to microalgae. So do you think we can, whatever waste is generated, the liquid part of the waste, can that be used for the production of seaweed or microalgae? Yeah. How can we grow seaweeds? If doesn't need any water, can it grow on wastewater then? Or how many of you know about microalgae? Cholera vulgaris, spirulina. Have you heard of spirulina? That is some places called as a superfood, yeah. So one of the aspect here is related to your microalgae, yeah. And with after this microalgae utilization, you will see how we go back to your how we can use the waste stream to grow microalgae. So it will be one step forward and then one step backward, yeah. So you will have to prepare an assignment which we will talk about that at the end of this particular lecture. So if you see microalgae, yeah, which is quite similar to macroalgae in terms of application, but is having lot more functions. So one is animal feed, pharmaceutical, nutraceutical application, your pesticides, biopesticides and other environmental chemicals, cosmetics and healthcare product, and textiles and industry applications like natural dye. Can you name any microalgae? Cholera vulgaris, yeah. Cholera vulgaris, spirulina, any other? Porphyridium. So these are all colored microalgae, yeah. They are colored because of phycocyanin. Phyco means color, cyanin means containing compounds, yeah. So they can impart color into the product. If you take a microalgae, add it to the milk, whosoever is fortifying it with vitamins of milk product, so you can have number of different colored milk product, yeah. Once you have colored milk product, then if you make a cheese out of it, then you will get a colored cheese, yeah. So why to have always white color paneer or white color cheese? So you will have a color, so you get both the functional properties of microalgae plus the protein from dairy, yeah. So there are a lot of different applications. You must be wondering what this microalgae is doing in quantification of waste and mapping the waste from aquatic food chain, yeah. So this is also one of the important biomass which is produced or which can be produced by using your wastewater. Yeah. So how these microalgae are grown? How do you grow these microalgae? So normally these microalgae, they needs an input of again same as whatever is required by plants. Yeah, CO2, nitrogen, fertilizer, all those nutrients plus optimum condition. Yeah, so there are plenty of different species which can be produced, and applications are very similar to macroalgae. Yeah. So we have, as I mentioned in the morning, we have a dedicated one for both macro and microalgae cultivation. And once you have a microalgae, one of the issue with the microalgae is amount of solid coming in liquid is not very high. Maximum you can get 2 grams solid per liter of water or so, or in some cases you can go up to 3 grams. So that means lot of, like whole the green water. If you keep a water in sun, you will see slowly it becomes green. Yeah. If you have water tank, it becomes green. It's just because of microalgae production. Yeah. That's why you try to close it with, you don't want to give access to the sunlight. Yeah. And that microalgae is basically what is now being used for the food purposes or for different applications. Yeah. And type of species you grow. So one of the aspect is you select those species growing on a wastewater, and then to cultivate that species and valorize the wastewater by growing microalgae. Yeah, that's why these things are very important, and in science observation is very important rather than knowing about things alone. Yeah, if you observe what is happening and then you can make it to your advantage. So if you'll see different application now. You saw the biorefinery approach for macroalgae, starting from production in the sea, or that macroalgae can also be done in fresh water. Similarly, in microalgae, if you select a strain on whatever wastewater it is growing or it can grow, suppose your objective is to absorb more of the CO2 from the environment or to absorb some of the nitrogen fertilizer, huge number of database or number of species are already there which can grow on different types of conditions. Whether it is less of nutrient, salt or pH. For example, you have whey liquid, whey is acidic, so you can still grow a microalgae on whey, but you have to select a strain which can grow on salt or at lower pH. Those who can retain a temperature or light stress, so whatever nutrients are required, and this is very well documented how you grow on different types of water. So the advantage here is if you grow it on a wastewater, so you can valorize the wastewater to recover the inorganic nutrient in a form nitrate, phosphate and potassium, and convert into organic form in a form of protein. So if you have a nitrate, NO2 or NO3 in a water, yeah, how this can be converted into proteins? By what process? That is again the plants. Plants, they absorb nutrient from nitrate, nitrites, and this is also a wastewater. Yeah. So that way you can convert into different nutrient, and once it is grown fully, you have to go for harvesting method, that is either by when you centrifuge it, you get all the cells accumulated at the bottom, yeah, and then water can be purified to certain stage, or use a flocculating agent, whatever you use it for your wastewater treatment, to see how you can precipitate the cells, and then recover those cells will become a microalgal cells. Yeah, it's very similar how we use it for any other process. Even when you are extracting a protein, you solubilize the protein in alkali, and then you precipitate the protein by using acid, by using isoelectric precipitation, and then you can go for extraction, cleaning, bioactivity and other quantification, whatever you have to do. Yeah, and then based on the species you have application for oil, protein, carbohydrate, vitamins, pigments and all those phenolics, biomass and everything. So this is complete biorefinery process. Whether you use a fresh water for specific purpose is the same way how the spirulina, cholera vulgaris, cholera sorkina, then you have purium porphyridium. There are number of different species are there which can be grown, and I will go into the details of different species when we go for microalgal production or how we can best valorize it. Yeah. So, this was one of those project which was between Interreg Atlantic area, that is Ireland and the west coast of UK, west coast of France and west coast of Spain. So, this is the organization which were involved in development of the whole process and how we can use the water to grow microalgae, and once microalgae is produced, how best you can use directly into the product. One application is to use the whole microalgae directly onto the product side of it. Extensive amount of research activities has been carried out already how we produce microalgae. What is limiting is how best we can make use of the product, developing a product, solving your health issues, wellness issues, another one, and delivering as a source of protein. Some microalgal species can have a protein as high as 52% on a dry basis. It's quite significant. Yeah. So, there you have to just recover the oil and then to have a protein. So, there are number of different areas what can be you can focus on. And in terms of mapping the wastewater and to recover the nutrient or to see the reduction of CO2, you can always measure how much CO2 a plant is consuming or how much CO2 a microalgae can consume over time. If I ask you a question, if how much of how to estimate CO2 consumption rate, how we can estimate how a plant can consume CO2. If suppose the environment CO2 is there, or if you take industrial emission, you have lot of CO2 coming out of it, yeah, of any process. How much CO2 it can consume? Suppose I was talking to one of your colleague yesterday, and we were discussing that if suppose there is CO2, take it as unit as a 2 liter of gas is there, yeah, and I have two options, plant A and plant B, yeah. It is grown in this particular environment. Which plant I should grow to capture maximum amount of CO2? How do we estimate that? We all know that banyan tree and peepal tree takes maximum CO2 and purifies the air. How this can be estimated? We all know that 6 CO2 plus 6 H2O, what it will produce? C6 H12 O6 plus oxygen, yeah. What is this process called? Photosynthesis. And we all studied when we were in primary schools, yeah. Not this equation, but the photosynthesis. But very same equation is even now it is valid. So, only plant can produce a glucose, and this glucose goes into starch as a storage material, and starch what we consume to get energy back in a form of glucose, and we store it in a form of glycogen, yeah, right. So, this is what the cycle, the way it works. So, if you go back to the carbon cycle or nitrogen cycle, same way coming back to the base question, if you have a tree A, that is how many of you like mango? All. Which one? Dashehri. So, take it as Dashehri. When I don't know how to write a spelling, I just make it like this, you know. And other mango variety is suppose Totapari, yeah, or Alfonso, yeah. Alfonso is used for mainly most of those mango drag. So, which tree will consume maximum amount of CO2 from environment? How do we know that? It's not rocket science question, it's not rocket science technique. So, any of the carbon dioxide which is absorbed, same thing applies for seaweed. When you saw the seaweed was there on a rope, it starts growing. When it grows, it collects a biomass, yeah. So, whatever biomass you have, if suppose biomass growth is 4 grams per day. Yeah, that means this 4 gram will have, you get an elemental analysis of carbon, hydrogen, oxygen, nitrogen, all this. So, based on this, carbon is coming from where? Only from CO2. Yeah, based on that you can determine how much CO2 it can absorb. Yeah, so that way you can develop what type of biomass should be used to recover that nutrient. Same way, if you measure the biomass, and if suppose coming back to microalgae, if microalgae is growing like this at this particular rate, at what point we should stop harvesting, or at what point you think is a maximum growth? You have to determine that, right? So, once you grow this biomass, you follow an equation, rate of biomass, that is per grams per day, and when it peaks up, and if you have a measured amount of biomass it is producing, based on that you can say the CO2 absorption, and similar pattern how much nitrate and nitrites are there in the water, yeah, and that is recovered or converted in a form of protein. Yeah, these are the two ways you can see how we can develop the production rate or extraction of nutrients from water in and conversion into the form of proteins. Yeah. So, this is the way we can establish the wastewater valorization. Yeah. So, that is one way of measuring that. Another application of these biomasses is to recovery of heavy metals. If you grow some of those plants, those heavy metals are recovered into the plant. That is another way of recovering those nutrients. It is very, very similar to the estimation of BOD and COD. Yeah. So, here BOD and COD you are measuring based on amount of oxygen it requires to oxidize the organic matter, but there is a chemical equation related to that. Very same case when you are trying to measure the BOD COD in a form of growing microalgae or by growing seaweeds. Yeah. So, this is what I was trying to cover on briefly about macroalgae, microalgae, and how these two are related to the wastewater. Now, what are the boundaries for the food waste? A general boundary, what we consider as a food waste, or how do we define a food waste? So, what is the difference between food loss or food waste? Very quick definition. The food loss which is happening all along, which can be used for food consumption. We saw yesterday all the statistics, how much it implies and what all it does. Whereas food waste is towards the end of consumer. So, if you will see pre-harvest losses, mature and unharvested left in a field, and then you have to go for a processing, yeah, processing losses, distribution and retail losses, and then consumption, that is again from household or for domestic level. So, this is the way we can estimate the food losses along a chain. Yeah. So, what is definition? This is definition coming which is adopted in Europe through Fusion. This was a large project, that is food waste all the way after the pre-harvesting all the way to consumer and household. Yeah. A. Other one is European Commission defines the food waste from the transportation side all the way to household. These are the boundary conditions for different definitions. That is also one of the reason we can change our statistics based on what we consider as a food waste or a food loss. Yeah. We just change the like suppose if you change a definition of food loss or food waste, then the values are changed. Yeah. This is also one of the phenomena. So, what is the definition to be used for food loss or food waste? You can define food waste in one way and you can define food waste in another way, but what is the universally accepted definition? We all know what is 1 meter. What is the 1 meter? No, that is correct. So 100 centimeter, very good. And then 1 centimeter is on a ruler, right? But if that ruler is in Kashmir, where the temperature is low, will have a metal contraction. Same 1 meter if it is in Middle East or near equator, the 1 meter will have linear expansion. Yeah. So what is exactly calibrated 1 meter? Same thing happens with the weight, 1 kg. Yeah. It's if somebody's weight is 80 kg, is not heavy, but he's on a wrong planet. If he goes on moon, weight will be different. Yeah. So there's a mass and a weight. Same way the length of a stuff. The important part is how do we define it. So before we say food losses are along agri-food chain is 30%, then you have to define where you calculate a food waste. The reason why I'm saying this one is in the seafood sector, in some cases there is zero food loss because fish is harvested, washed, goes to the domestic one. So there is always everything related to food waste at the consumer end. Do you agree? Yeah. Whereas if the fish is processed commercially to produce a fillet, then you have a food loss plus food waste. In the processed product, food waste is less, whereas in processed product, food loss is there and food waste is there, whereas when it comes to whole fish consumption, only food waste is there. Did you get my point? Yeah. So how do we define that? So if you take United Nations Sustainability Developmental Goals again, all those 17 different goals. So this is the way food waste and food loss is defined in relation to Sustainability Developmental Goal number 12. Number 12 is related to what? Responsible consumption and production. So you have pre-harvest activity. So when it comes to pre-harvest activity, what are those related to seafood? Aquaculture or harvesting? Yeah. So you don't have any of those pre-harvest activities when it comes to seafood. These activities are applicable for terrestrial or land-based crops like pulses and cereals and other grains. And then you have harvesting. This is quite common in every agri-food operations. Then you have on-farm operation in terms of fish. What is it? Seafood, what it will be? That is your washing, cleaning and keeping it in freezing condition, right? Yeah. And then you have a transportation, storage and distribution, processing and packaging before freezing or live material, live biomass. Now when it comes to fish, what could be the food losses along a chain? Seafood. Guess. Whether it's an aquaculture or any other thing, what could be the food losses along the chain from harvesting till this point before retail? So you just think, you imagine a situation, you go get 10 kg of fish from the sea, yeah, you wash it, you pack it. When you get 10 kg of fish, so what could be the losses before you freeze it or thaw it? Because this is the topic what we are going to be, this lecture and the next one, more it will be, there is no fundamental principles behind, there are fundamental principles are there, application of those all depends upon majority in a form of common sense. Yeah, there cannot be that you can valorize a waste in a specific manner. We all have a different eating habit, we all have different product development strategies. So based on that, waste generates. So what could be the waste on the fish again, I am not talking about the retail or consumption.
Fin will be there, fish fin,
I am
inedible
talking about
offals.
whole fish. That is processing side of it, right? Yeah, correct. Fins, scales and all those are the waste. I am talking about the whole fish. What could be the waste? You throw a net, you go with a big boat in a sea, throws a net, you harvest a fish, yeah, or even aquaculture fish is harvested. So what could be the waste there? A is damaged fish, correct? B is spoiled or half killed fish, yeah, or whatever damage is there, or diseased fish, yeah, correct? It can have a disease. And C,
Inedible fishes, like
that is bycatches, yeah, right? That means smaller fish or larger fish which are picked along with it. When it comes to mussels or shellfish, it could be a damaged shells, because some shells are bought directly as a whole, yeah, diseased or damaged. That means you use a visual inspection to segregate it. So how you can avoid that one is what will be the valorization strategies for those. So that type of species can go for fertilizer or for different application. It's very similar if you harvest apples. All apples which are sound, that can go as a table apple, which are having bruises and all, that can go for jam production or jellies production or making for juice production strategy. Similar thing applies for seafood also. There is only the drawing the parallels from the other processing streams. Now when it comes to food waste, what it could be again for the food waste? So processing and packaging we will be covering in subsequent sections, like all you get fins, guts, viscera, and some cases in order to digest or to break the microalgae or macroalgae species from enzyme, you can always recover the enzyme from the gut of fish, because they have those enzymes which eats seaweeds, yeah, as well as microalgae which is grown in the water. So how best you can use that particular enzyme extraction from the fish viscera to use it as an enzymatic processes, yeah. That's one easy way of doing it. And all these damaged, spoiled, diseased fish or bycatches, where you can use this? What could be the application? Fertilizer already discussed, feed already discussed to some extent, yeah. What could be another application? BAIT, yeah, correct. So you can use those fish to attract other fish to catch it into your net or other specific one, yeah. So it's not somewhere it has been investigated, research is done. It is just you thought, oh, it will be good to use it as a bait, rather than using a whole fish as a baiting purpose, yeah. So that's that's one. So when it comes to sustainability developmental goal, 12.3.1 is considered from harvesting till processing and packaging. 12.3.2 is from when it is processed, that is at retail and consumption level, yeah. So whenever you are defining waste or valorizing waste, it is very important to establish a boundary, yeah. Otherwise it becomes meaningless. You may estimate a waste in a specific quantity, and another colleague of yours will say, in my process I have less amount of waste, just because where you are drawing the boundaries, yeah. So when it comes to these type of waste, if you are able to address those different type of waste or losses, we will be able to address clearly number of different United Nations developmental goals, yeah. So how we can reduce food losses or food waste what will be the strategy? How we can reduce our food losses during the transportation? Cold chain is one of the most common answer everywhere it has been seen, yeah, and is common for every biomass or every crop, yeah. What could be the alternative to the cold chain, yeah? So that is any solutions related to that? I know cold chain is there, potatoes are there, there is always a cold chain required. Wheat is there, always cold chain is required. But what for? Think of the time when there were no refrigerators, how those were preserved, yeah? Evaporative cooling, yeah? Or digging the soil and putting it over there, yeah? These are the traditional methods. Are there any other technique you can preserve those type of element? Yeah. So this is all the thinking process. Sometimes we learn a lot from our history or from class 1 preservation, which can be now slightly optimized or can be advanced. So if we are able to manage food loss and waste, that is no poverty, zero hunger, good health and well-being, and affordable and clean energy, responsible production and consumption, climate action, life on land, yeah. This is applicable also for your seafood, or is only applicable for seafood, because once it is in the water, you have taken it out, then you are talking about land. So all nutraceutical storage and other. So all these are the ways you can draw out your waste strategy or what could be the advantages, yeah. So as you can see, this is all some of the knowledge and whatever been talking based on the experience working with different industry partners, working lot with masters and undergrad students who come in for a placement. They come up with lot of different new ideas which we nurture it and come up with the process, yeah. It's not that all the scientific knowledge and everything whatever your supervisors or lecturer is telling, they know everything, no, yeah. That's the way it builds up the knowledge bank and we progress further, yeah. So food losses and side stream when it comes to edible aquatic product. So here this is a broad one calculated and obtained from a specific region. So wild capture, as you can see, this is a boat, have a bit net, so you get everything collected. Your jellyfish, sponge, you went for a fish, you buy everything. Something like you throw a net, that means you enter into a supermarket, you went in to buy a specific product, but you came with 5 shopping bags, yeah. So this is one. On land processing, that means how you are processing it or segregating it or removing whatever you don't need, that's on land. Retail, food services, that is HORAC is hospitality and restaurant and catering businesses, yeah, because everything doesn't go to the straight to household. There are a lot of consumption are also done in food restaurants, catering businesses, or there is a party they need 200 kg of mussels. So all those element comes under food services, and then you have a household consumption. So you can see where all we have losses along the chain, yeah. So that means approximately this many tons. So these are some of the global figures, but everything whatever is there goes to bin, which is again not advisable because we are losing good resources into the bin, whether it goes for landfill or goes for incineration, yeah. So what are the approaches, how we can reduce or quantify the waste and food losses, or what are the strategy, yeah. Only 2 strategies, simple. Reduce your food waste and food losses or transform that food losses and food waste, correct? It's very simple, isn't it? But how can we reduce food losses? Reduction of food losses and food waste along, I keep forgetting this is a pen, you can rub it from the other side, along agri food chain, yeah, or whatever waste is generated. So, how that waste can be transformed, yeah. Any idea? Any other technique to reduce waste? So, there could be 100 different ways of reducing waste and transforming the waste. Do you think any product which does not contain any waste, because a lot of commercial companies they claim their production process does not generate any waste. Is it possible? There is always avoidable waste, yeah, and unavoidable. You take any of the crop, there will be always the waste which can be avoided, or there is a product which you cannot avoid, there will be a waste generated. So, how we can valorize unavoidable waste scenario? For example, the whole fish itself, you won't be eating the visceral organ, or you won't be eating this skin or shells. Same way, if you take a plant crop, wheat grain can be consumed with the bran or the whole meal flour for making chapatis and other product. You have refined wheat flour making breads and cookies and other thing. Hull and straw. It is farmers, they know how to make use of it, yeah. Same thing, if you have mussels, you have shells, where you can use those shells. The shells they have, I am not talking about chitin, chitosan and all those high value ingredient peptides and everything, that is all at the research level, or it is a specialized sectors are required. For that good scientific knowledge is required, which you guys have. Think of the farmer, how he can use those shells. Crush those shells, use it as a source of calcium, or to use it for making your along with the cement and all to make houses. If you go on the coastal areas, especially near about Rameshwaram and other places, you will see how best uses are done. Even those shells which are cleaned, and people they keep it in their house as a decoration or making all those curtains and other things, yeah. So, these are also some of the application how it can be used. Traditionally in even in Uttar Pradesh, all those shells were used to peel potatoes. How many of you have seen it? Yeah? My mother used to use those shells to peel the potatoes. Like, you know, use peeling potatoes, you cut in between and use it for peeling. So, there are different applications are there. So, all these things are quite useful. Nowadays, for the sake of being in a nature, natural way, we can reduce or valorize that waste, yeah. So, in this case, you have to have 3 or 4 different people sit down together and see what are the waste generated and how you can make use of those waste streams, yeah. So, since we all are scientists or we all are studying science, so we always try to come up with a complicated solutions, yeah. So, how we can reduce, that is the main focus is to improving the microbial safety, yeah, and processing efficiency, yeah. So, when it comes to microbial safety, that means how we can reduce the energy and water usage. You mentioned about the freezing chain, I am just picking that particular point, and that requires huge amount of energy. Or for washing, how we can avoid usage of water to wash those side streams or wash the whole biomass or side stream, and how we can reduce waste and how we can create a new applications, yeah. That is all comes under reduction strategy. I am trying to simplify as much as I can, yeah, and And this is quite simple. You want to reduce a waste? What is the strategy? Why you want to reduce a waste, or how we can do it? A, by ensuring a microbial safety. If you are safe, then only you are talking about the quantity and the quality and other attributes, and improving process efficiency. So, energy of water reduction, waste, and creating new application of those streams so that you can reduce the wastage. Whether it is, I will be using interchangeably both food losses and food waste. Yeah. Now, how many of you have heard about new technologies? Yep. Now, this is basically all the technologies which can be used for advanced processing as an alternative to thermal processing. Yeah. Don't get shocked with seeing all this high pressure, pulse electric field, ultrasound, cold plasma, lipid oxidation, proteolysis, oxidation. Yeah. We will be going back to these technologies again when we go for transformational strategies, right? What it shows about high pressure, many of you are or from undergrad, masters and a PhD. High pressure processing is very similar to pressure cooking. Yeah. Only difference is pressure used is of more than 200 to 600 megapascals, and some places you will see 800, 900 megapascals. But commercially, anything more than 600 megapascals is quite challenging because of strength of material and requirement. Yeah. So these are known as non-thermal technologies. Why we call it non-thermal?
So using low temperature technology.
Correct. But low temperature, yeah, you all said about low temperature, right? But you know microorganisms can only be killed at 70 degree or 72 degrees Celsius. Yeah. Water boils at what temperature?
100 degree.
Can water boil at 90 degrees Celsius?
Yes, yes. High.
Correct. If you go on mountain. Why I quoted this one? Because this is what we studied in our school. Yeah. When you go on a mountain, water boils at lower temperature. Yeah. So that means pressure is higher. Yeah. But if I reduce the pressure, yeah, or sorry, if I increase the pressure, temperature will boil at lower pressure. Yeah. If you know PV is equal to nRT. P is pressure, V is voltage. Good. Yeah, all are awake. That's nice. N is your mole, R is universal gas constant, T is temperature. Yeah. So the water which boils at 100 degrees Celsius is normal atmospheric pressure, that is 1 bar. Yeah. If you increase the pressure to 5000 bar, what will happen? Same thermal effect is observed at sublethal temperature. That's the only definition of non-thermal technologies. Yeah. Achieving microbial safety at sublethal temperature is non-thermal. Yeah. Because temperature is always increasing. Yeah. Temperature will get those application. Same thing ultrasound, your friend here is working on thermosonication using ultrasonic cavitation. Cold plasma you have already seen. Yeah. You all saw the cold plasma, right? Yeah. What way cold plasma works? Yeah. Because you have seen the practical other day, that's the reason I'm asking you. Do the guess, whatever you learned from there, that's fine.
Sir, the cold plasma is working to the high voltage is applied in closed chamber. The fourth stage of matter is like glow, like matter is produced. That glow is create the radicals, free radicals in the food. Like due to the presence of moisture in food, the glow is produce the free radicals in the food. So it is toxic to the microorganism. It only used for a surface treatment for the foods.
Okay, that's correct, and I'll make it very simple because I always like to know things in a very base language. Yeah, plasma is a fourth state of matter. Yeah, if you have a solid, if you heat, what will happen? It converts into liquid. Yeah, if you further heat it, it becomes what? Gas. Yeah, and if you further breaks the gas, what it will form? Ions. Yeah, so that means O2, if it is broken down, it will form O minus. Yeah, these are all free radicals, right? And these are free radical, so they are ready to attack anything because everybody want to get stable. Yeah, it's same as with us. You want to study and then you want to get what is the common word used when you are going to settle in a life. Yeah, everybody need to get settled. It takes time for different radical. So when this is a free radical, it will try to stabilize. Their transient time is very small. Yeah, so they will look for oxidizing the organic matter or whatever comes on their way. Yeah, and this, to create this free radical, you need very high voltage. That's one, or very high temperature. Yeah, either you need very high temperature or you need high voltage. So voltage can be in the tune of suppose 100 kVA. Yeah, but do you think if you use 100 kVA or 10 kVA or 12 kVA, what will happen to human being? But at the same time, plasma is also used for cancer research. We do lot of work on plasma for targeted delivery for tumor to create holes on a tumor through by plasma, so that we can deliver a targeted drugs. So can you explain, if you are talking about such a high voltage to create a plasma, even 10 kVA is okay for making plasma. Such a 10 kilovolt voltage, and you are generating it from 220 volt, what you are getting it, right? There is no substation in that plasma system. So still you are not getting electric shock. Remember, it's not the voltage which is important. Important is the current. Yeah. So if voltage is 12 volt and 5 amps, if we increase the voltage to 12,000 ampere becomes 0.1 amps or so. So you won't get an electric shock, but this voltage is helpful in creating those species. Yeah. So those free radical which are created, they cause mainly oxidation. Yeah. So one of the key inactivation mechanism for microorganism is by oxidation. Same thing happens with the chlorine. When we keep saying that chlorine is used for inactivation, how chlorine works? To generate a chlorine one, we use sodium hypochlorite. Yeah, that is NaOCl. Yeah, this is the one nascent oxygen which causes microbial inactivation. Make sense? Yeah. So everywhere, if you know the mechanism, then you can deploy that. Yeah. That's why whenever free radical is formed, you have a water, it forms acidic solution. Yeah. Intentionally, I did not put a slide which talks about different technology and microbial inactivation. Yeah. So when we go for the further different technologies, there you will see how these technologies can inactivate microorganisms, and then you can relate this chemistry with microbial one. Yeah. So high pressure, that is by rupturing. Yeah, because of pressure it ruptures the cell. Ultrasonic, it forms, what is cavitation? Yeah, good. Yeah.
So that ultrasound creates a cavitation in the form of pressure difference like rarefaction and compression. By this difference, the microbial inactivation has been done, sir.
Okay. So how ultrasonic works basically, it forms compression, rarefaction and all. Somebody is asking you, please do the work. You won't listen. Somebody increase the frequency. Do the work. You start doing it, yeah? That's the way sound works, yeah?
Yes.
The simple way. Ultrasonic, when it passes through water, it forms cavities, yeah? These cavities are cavitating bubbles. Either they explode or they implode, yeah? That is cavitation formed because of sound pressure, yeah? But you can also create a cavities based on water. If you turn on a tap, you see the water coming out very easily, yeah? If you increase the speed of the water and the outlet of the water is the same diameter tap, it creates a lot of bubbles, yeah? These are cavities. They break due to their internal pressure, that is implode or explode. During that process, it also breaks the water into their free radicals, yeah? And this creates sonoporation on microorganisms or also causes breakdown of different molecules, yeah? So that is the application, and that cavity formation depends upon the frequency of ultrasound, yeah?
Yes, sir.
What is the frequency of ultrasonic system?
So 20 to 40 kilohertz, it may be changed.
20 kilohertz, yeah?
To 20 megahertz. 2 megahertz. So basically, in ultrasound bath, 20 to 40 kilohertz has been in usage, sir.
Okay. Now, when it comes to bath system or a probe system, anything, you need a piezoelectric transducer. What is piezoelectricity? Electricity, when you press it, it forms electricity. Can you know the piezoelectric material which you all might have it?
Quartz.
Correct. So quartz is having an ability, when you press it, it distributes positive and negative, and that creates an electric current. So that's the way watch, they all have a quartz as a piezoelectric material. Same way, when you give an electric current, those charges, they generate a pressure, yeah? That's the principle used for ultrasonic probes, yeah? Now, when it comes to ultrasound, the frequency range anything about 20 kilohertz, yeah, all the way to even 10 megahertz. Anything above 5 megahertz is used mainly for diagnostic purpose, car sensors and everything, and processing zone between 20 to close to 1 megahertz depending upon the application. When we'll go through the technology, I'll show you number of different types of system. What you have seen is what is commercially most prominently available. But there are 400 kilohertz, 600 kilohertz, and there is no relationship between what is best at 20 kilohertz may not work at 100 kilohertz, what works at 100 kilohertz may not work at 20 kilohertz, yeah? So it's always cause and effects. That's why ultrasound itself is an area of intense research. And then you have a pulse electric field. Here also we are talking about high amount of voltage, yeah, but along with the frequency of the current and frequency as well as on the current. So this causes electroporation, that means giving electric shock. So what happens, these are known for improving the microbial safety, but they do cause changes in protein and lipids, mainly oxidation and what? Oxidation. Protein gets oxidation and lipids get oxidized, yeah? You would have seen, heard a term called protein denaturation happens, yeah? But do you think protein denaturation is good or a bad? Okay. How it will be good?
For improvement of texture and all, this protein denaturation will be like in a positive manner, and in terms of nutrition, it is a negative impact.
Okay. So what is basically a denaturation?
Change of structure in the proteins.
correct? Then what type of structure? They have 4 different stages structure, right?
Primary, secondary, tertiary.
We'll start with the bigger one. Quaternary structure, tertiary structure, secondary structure and primary structure. So what is the primary structure is? Sequence of amino acids, yeah? So in body we need to break a primary structure in order to absorb amino acids, yeah? And this amino acid attached with the peptide bond like a rope, yeah? And then this rope are staked on different layers, yeah? That is your secondary structure, yeah? And those different ropes are structured into a coil form or different way. Then it becomes a tertiary structure. I am not going to go into primary structure, like into the protein chemistry and all. And this tertiary structure, when it is joined together, makes a quaternary structure. So even if you shake a mill, you get denaturation, yeah? And we have to denature to improve your digestibility, bioavailability and everything, correct? Raw wheat flour, if you consume, you are done with the stomach upset. If you roast it, you make it panjiri or you make a laddu, yeah? So you get a different flavor. So denaturation is important, but if you are looking for a specific application for gel, hydrogel, aerating gel or gluten network, there you don't need denaturation, yeah? So commonly saying denaturation is not good is not a correct statement, yeah? Depends upon where do we need it. So in these proteins and lipid, all these things, they do have impact of all the technologies. How this impact can be changed depending upon the condition we need it to use. Cold plasma, that is one of the way where you can use plasma activated water, that means a free radical in a water having a fish in it can improve the shelf life on farm, yeah? Do you agree with that? So that is the way how you can deploy different techniques. So if you see here, second aspect is transformation. So we are not going into different technologies, how we can reduce the food waste along the processing by deploying different non-thermal technologies, thermal processing and all. If we go into that, we will deviate ourselves into completely different domain. Other one is biotransformation. That means whatever biomass we have, whether it is a waste, whether we can add value to it or a whole biomass, we can convert into zero waste refineries. The example I was quoting only related to seaweed and microalgae because these are relatively new biomass. So let us do first thing right itself. That means design a process where you have every fraction is utilized, yeah? Because this concept was not there 20 years, 30 years ago when we started consuming fish or shells. We just was only interested in the grains, forget about it. You know, in northern state, the straw is having very good functions, but what we do to have a quick turnaround, we burn it in a field, yeah? Still you get some minerals and you want to get the next crop. But if you have a process to make the maximum value out of a straw, in summer straw is not available, in winter it is available, yeah? The straws which is required for animal feed, that is not available in summer because all the animals, they are starving because they need something to eat. So if you preserve the straw over 3-4 months, you make value to it and at the same time you solve the feeding problem for cattle, yeah? And they require simple interventions. You don't need a rocket science technology to do that, yeah? Okay, again bit of deviation when it comes to straw. How do we preserve bread? By using calcium propionate, yeah? Very same calcium propionate which avoids the mold and fungal growth. If it is added with the straw and packed, straw can be kept for 5 to 6 months. There are multiple companies who are making this type of medicated straw which is also used for making animal bedding. And in some cases that particular straw, what is added in is cow urine or any of those byproducts so that animal won't eat that which is there used for their bedding purposes, yeah? So these are the simple intervention which I have seen people doing it on a farm. Not only in Europe, even in India they do have their own way, but that requires some bit of brushing up with the knowledge and technology and to make into a full-fledged process. Yeah. Now coming back to this biotransformation, again related to sustainability developmental goals. Here land under the water comes in, climate action, responsible consumption, infrastructure and good health and well-being. So it is related to different sustainability developmental goals. So what are the transformational strategies? Yeah. So very same technologies, what you see here, which are what we discussed briefly about non-thermal technologies or new processing, new pretreatment technologies and novel extraction, which can be used for improving shelf life and safety. Yeah. Enhanced recovery. So if you use ultrasonics for preservation, you can also use ultrasonic for extraction purpose. Yeah. Because it is disrupting a matrix. High pressure treatment will disrupt the biomass so that you can have better protein recovery or better nutrient recovery. And when you use all these new technologies, you have a better nutritional properties compared to conventional method as a thermal processing. You would have seen a lot in your lectures about non-thermal technology, so I am not going to go into details of it. And I work mainly on range of different non-thermal technologies, and if you have any question related to that pertinent to this particular module or any other, feel free to ask. Yeah. And then once we are able to use this technology either with the processing, pre-treatment or for extraction, we can add value and we can utilize waste. Yeah. I can quote number of different technologies, number of different applications to valorize different processes. Now, this will take bit of time to digest. Yeah. This is from a reference of 2020. If you want to know about the paper, I can, and this is pretty much clear, you can read it. Yeah. This is a high resolution photograph. What it shows, what are the food waste, yeah, from different streams and what are the potential pathways you can generate. How many of you have seen this publication? So you will see this particular specific reference of publication in majority the report which are dealing with the food waste, food losses, valorization. So if you will see fish based product, these blue lines, if you track these blue lines, what all you can get? Fatty acids, yeah, enzymes, minerals, yeah, and then cereals, see this line, yeah, okay. That's the way to read it. Half is your food waste and half is our different pathway, and all these ones are different application, whether to use it agriculture, pharmaceutical, medical sector, food industry, biomaterial, textiles, pulp and paper, environmental sector and chemical industry. Yeah? Make sense? So, you can make this particular diagram. These are potential application how this can be valorized for different pathways. Yeah? So, this is all your fish. Is there any crop which is missing from here? You have seafood, you have dairy, cereals, fruits, meat, oilseed, sugar, starchy crop, vegetables. Anything else which is missing? I know there is something missing there. Yeah? Somewhat it is related in seafood. It is, this is partially missing. This is partially missing, and completely missing is seaweed, yeah, and microalgae, yeah. So majority of the focus I'll try to shift it on the plant side of it, yeah, because that information is limited in the literature, and rest all valorization strategies you can easily find into different different literature. So with this thought, what we are quantifying, we need to quantify all the waste, whatever is coming in, and how we can valorize it for different application in human food or feed applications, yeah. So what we have covered so far? Actually, when I'm seeing somebody, they're just putting their head down, yeah. So don't worry, I'm not going to pinpoint anybody. You know, it's just as I already said, we do a lot of mistakes. You guys do mistakes, and we learn from each other, yeah. So basically, what we are trying to cover was to do with the biorefinery approach, yeah, so that we can make use of all the application. And in order to do that, we need to quantify the waste, yeah, and how we can reduce or transform the waste, yeah, okay. So in next lecture, we'll be covering bit on the quantification of waste and streams, and going back how we can reduce it at a consumer level or at retail or at a processing level, yeah. So remember the definition of food loss and food waste, they both comes in umbrella of UN SDG number 12, that is responsible production and consumption, yeah. That's great. So we'll start maybe after 15 minutes break. So far we were discussing about the waste and everything. So since it was divided into 3 different sections, one was quantification of waste from farm to fork, and then valorization and how we can reduce. So when I say reduce wastage at consumer level, who is actually a consumer? Consumer for the primary producer is a secondary processor, and for secondary or tertiary processor, consumer is human, that is household, yeah. Consumer does not mean all, we can put in a simple word called end user, yeah, who is using at the end of a processing chain, yeah. So ultimate end user is human for consumption purpose, yeah. But if it is made as a fertilizer, end user becomes the plants, yeah. You know, so that's the way it can be looked into. So, so what we were looking into about the biorefinery, how we can create stuff. So something is already well established. Seafood processing, squids, mollusks, shells is very well established. So how we can use those resources in a better way, using better what we already use. If you remember that big bioeconomy diagram, the top portion it says using better what we already use, yeah, that is applicable to existing seafood, or unlocking the potential of seas and ocean at the bottom one, yeah. So main focus is at the bottom one, because this is the new biomass which is not very popular in this region as well as in Europe, whereas this is yet to be explored because a lot of different species are there, lot of different biomasses which we are not consuming, which we are not using, but they have a huge potential for different applications. Yeah, because on earth we have almost 70% water. Yeah. Now, what could be the valorization strategy? So, if you look at this particular seafood, what we generate is both solid side stream and liquid side stream. That will be the focus of tomorrow's lecture, valorization strategies for solid side stream or liquid side stream. So, liquid side stream is what we get as a liquid side stream, mainly related to water, processing water, or the water which is used for extraction purposes or for cleaning purposes. And what other liquid side stream? Can you guess? Blood. Yeah, fish also contains blood, isn't it? Yeah, think of the bigger one. Yeah, big fish, they also contains the blood. Yeah, so how that blood can also be valorized? The same thing applies for animal side of it. There is a huge amount of blood, whether it's halal process or a jhatka process, whatever way it is. So there is a blood, it has to be utilized some way or the other. Same thing applies also from the fish side of it. Yeah, some fish is consumed as a whole, and blood also contains all the nutrients, minerals and iron and the protein. So how that can be valorized? So this is all the liquid side stream. So water, whatever is coming out of the processing, will also have some elements of blood. So that's why they have high amount of biological oxygen demand and chemical oxygen demand. So here fish, one waste is fish skin. Yeah, I have seen some people removing the fish skin by putting hot water on a salmon, the skin is removed. Am I correct in saying that? Yeah, that's very easy to remove that skin, and that skin is rich in collagen. Yeah. Other one is fish muscle, the head or a tail. Yeah, or removing the scales, which is done at a domestic level also, at a processing level also. Yeah, when you go for retail or processing, there is a huge amount of biomass is collected. And then you have a fish frame, that is bones and scales, whatever is left over, or frame and bones anyway, the structure and all those bone stuff. So where we can use these bones? Fish bones. Yeah, you will have to think yourself where we can use these bones. Yeah,
amount of calcium it is having.
Correct, calcium extraction. What else? Have you seen the bone meal? Yeah, go on. Yeah, calcium and
I was about to say calciums.
Yeah, correct. Yeah, calcium is a good source, bone is good source of calcium. They also have a good amount of protein. Yeah, that protein is having a structural protein, and peptides obtained from such bones are very good for joint health and other one, because they have those properties. Yeah. Second one is, if you heat all the bones to 900 to 1200 degrees Celsius, yeah, what we get? Crystalline structure of calcium, and that is, have you heard this word, hydroxy apatite? Have you heard this material? Yeah, this is a amorphous form of calcium which is used to make a prosthetic legs and different structure. Yeah, so that is another way how this type of minerals of calcium and phosphorus can be used for hydroxyapatite. So I am not going to go into the process of that. You can look into your, you all are good in looking into the internet anyway. Yeah, just type in the word hydroxyapatite, you will see any type of bones can be converted into that application. And that is, fish bones will have a different properties compared to hydroxyapatite formed from animal bones, or forget about the human bones anyway, because human bone goes to Ganga or in ashes. Yeah. So basically ashing, that's minerals, and the minerals form amorphous structure. Yeah. It's very similar when we have okay, bit of deviation, the glass, yeah, normal glass, how it is made?
Silica.
Silica means sand,
Sand.
yeah, sand structure is correct, SiO4. So, if you heat a sand, yeah, to what temperature you get a glass? It has to cross more than 2000 degrees Celsius, yeah, to melt silica, yeah, and then it is cooled, then you get a glassy structure. Those who have studied the plant food processing, you know starch glassy structure. You heat a starch is viscous, is translucent. When you heat it, it becomes slightly transparent, yeah, like a transparent gel. In terminology, in rheology, we use called glass transition stage, yeah, that makes a glassy transition. So, silica is nothing but supercooled liquid made up of silicon, yeah. So, that is what way silicon structure is changed to make a glassy structure, yeah. Same glass it is melted to make molding. If you go in any of those places where they make bottles and glasses and other ones, so this is the simple procedure used. Very similar one, when you have this calcium and other metals, when you heat it to a certain temperature, either you make a biochar, you make an activated carbon, that means to make a carbon in reduced oxygen. So, it becomes black, and then you acidify it to make a activated carbon which can be used for filtration purposes. A, heat the shells or the bones to a higher temperature and it makes an amorphous structure, and when you try to filter it, it will help in filtration of water, trapment of microorganisms in a water, those type of filters which we use it in drinking water facilities, yeah. So, these are some of the non-food applications of it, yeah. So, we can have both food or agri-food sector. When we say agri-food sector, allied sector, because food is in the center, chemical engineer can become a food scientist, mechanical engineer can become a food scientist, but food scientist has to remain in food science, yeah. And the phrase what I use in first lecture, food science is a common science which is not so common in common people, yeah. So, majority of the operation comes from the common sense. Now, your crab shells, prawn shells, lobster shells, shrimp shells, and to your benefit, some of the research scholar of NIT, they have collected the shells here to do some of the practical here for you purpose. So, we should be thankful to them, and we possibly will do maybe tomorrow, yeah, the practical to see how we make use of these shells, yeah, how do we dissolve these shells, yeah. So, that will cover once you do the experiment, see the experiment, and then it becomes easier to apply, yeah. Then other one is your liquid side stream, bleeding water, that is blood, processing water, wastewater during cleaning and rinsing operations, that includes the vessel cleaning as well as cleaning of the biomass, cooking and blanching liquid, because you use sometimes cooked fish for salting and oil purposes, sanitary waste. So, all this water goes as wastewater solid and wastewater dissolved nutrients, yeah. So, if you are trying to recover a food material, but the problem is how do you segregate the waste, yeah. Cleaning water is one, washing of fruit or washing of boiled potato is a potable water, whereas water used to clean the furs or the processing equipment with the chlorine and all is wash water, that is water used for cleaning purposes, and all the water in industries mixed together goes to wastewater treatment. The water which is used to wash the fish fillet or washing of the product which is for human food consumption, it is still a potable water, that means it is having all the nutrient. So, if you have a clear segregation of the water, you can use that water for food ingredients, yeah. The moment it is mixed together, then it will have to go through a certain regulation to be used for human food, yeah. And there can be easily a debate if you use a wastewater to grow a crop or to grow certain say duckweed, seaweed or microalgae, can it be qualified as a food ingredient or not? So, that means if there is a tree which gets a rainwater and there is another tree which gets a sewer water, yeah, to grow a fruit, will that fruit will be considered as a good fruit or a bad fruit? Yeah, you got my point. Yeah, because ultimately all those animal feces and all are used for biofertilizer that grows into the crop. So, that is all has to go through a specific regulation depending upon what type of water is used for the production. That is also important. It used to be important how you produce a food, but in what way it is processed or produced is also equally important. You want to become a rich, but how you become a rich is also very important. Yeah, you are going through a green economy or you are going through a black economy. Yeah, so getting the product and how it is being processed, they are equally important for traceability point of view and based on socio-cultural attributes. So, I am not going to go into that. Then you have different extraction technologies. You got a brief flavor of some of the technologies, whether it is a chemical, acid alkali, deacetylation, that is again for converting chitin into chitosan, fermentation using microorganisms, that means there are certain microorganisms which can also dissolve shells, enzymatic hydrolysis, that is enzymes which can break those proteins into peptides, or chitin into chitinase, or enzyme which breaks the chitin to release a protein, or enzyme which breaks a protein to release your chitin. Yeah. Other techniques, supercritical fluid extraction. How many of you know about supercritical fluid extraction? Yeah, go on, yeah, tell.
I know about like CO2 as a supercritical fluid. It used to extract the bioactive compounds from some plants and food matters. That extraction, the CO2 is in both levels, like liquid and gas in same time. The high pressure and high temperature used to extract the compound from the food materials.
Correct. So, supercritical or a subcritical stage. So, how many of you use perfume or deodorants? Everybody, right? It is a liquid inside. Yeah. Even LPG gas is a liquid. Yeah. The moment we release the pressure, it becomes a gas. Yeah. Same way the CO2, if you pressurize it, becomes liquid CO2. Yeah. And the liquid CO2 is useful for extracting non-polar compound. If you have to extract water soluble vitamins, that is, you need just a water as a solvent. Yeah. When you have to get the fat soluble vitamins, so what do you need? You need a non-polar solvent. So, most used non-polar solvent is hexane. Yeah. But when we use hexane as a solvent, the hexane residues are left over. Yeah. How we can get rid of hexane? What is the natural non-polar solvent which can extract your oil? If you have 2% oil or 3% oil in fish biomass left over, that also contains carotenoids, astaxanthin as some of the bioactives which are soluble in non-polar, non-water solvent. What is an alternative to hexane? Yeah. Don't worry, no petroleum ether, diethyl ether and all those are chemicals. Yeah. Hexane is much better than food grade hexane and all. Alternative to any of those chemicals, how we can get a natural one? See, diethyl ether, ester and petroleum ether and hexane, they all are organic solvents. Yeah. How we get rid of organic solvent to make a process completely green? CO2 is there, right? At what pressure and conditions CO2 becomes a liquid? That becomes a chemistry question. If you pressurize a CO2, it is a liquid, and then in same pressurized condition, if it is mixed with the biomass, all the non-polar will be dissolved in liquid CO2. You take that liquid CO2, release the pressure, CO2 is gone and you get your product. Yeah, that's the simple principle of supercritical. Yeah. Apart from supercritical CO2, is there any way we can get, suppose if I want to get astaxanthin out of shells or the shrimp shells or lobster shells, to get that bioactive into a oil? Is there any way we can get that particular one without using hexane or supercritical? Any idea? Come up with a creative one, at least we'll learn something.
Deep eutatic solvent extraction.
Deep eutatic, that means having a mixture 2-3 different solvent to get this one, right? So, if suppose I have say for example coconut, coconut oil, yeah, and I want to make it a healthy oil, yeah, it itself is healthy. If you want to add astaxanthin or from shrimp shells into it, what will be the best way? One is to extract it and to add into this oil, yeah. What could be other technique? Yes, that's good.
Microwave assisted extractions, because a particular frequency can extract a particular compound from the mass.
Correct. One is solvent free extraction, yeah. How many of you have made ghee, that's called desi ghee, out of butter? If you take a milk, you have made it, yeah. How you made it? How you make it?
Sir,
Go on, yeah.
by applying heat, sir.
When you heat it, if you have seen, if you have a milk, you freeze a full fat milk and thaw it, and the moment you heat it, you will see the fat droplets on top, yeah. That means fat is present in a biomass. If you heat it, as your colleague mentioned about microwave, it will separate the lipids, yeah, because that milk is basically oil in water emulsion, yeah. So, you can break that emulsion by heating or so, yeah. So, one of the way is, that's same thing with ultrasonics as well, same thing with RF heating, ohmic heating, or just normal heating. How we make butter? If you have a cream, you heat a cream, slowly all that oil is separated and you get your protein completely precipitated or denatured. That is the way ghee is prepared, yeah. So, in a similar fashion, with the same biomass, if it is cooked and with whatever technique, whether novel thermal or novel non-thermal technique, and if you squeeze it, you can get oil clearly separated, yeah. And then it comes for deodorization, degumming, all those processes to get refined oil, yeah. Whereas virgin oil is directly extracted. So, you have to see the coconut oil extraction. We all eat copra, yeah. We all eat coconut water. Same coconut is also used to get virgin coconut parachute one, yeah. So, how that coconut oil is prepared? Think the similar way is a fish biomass, how we can get oil out of it, yeah. So, that is what I keep saying, not only here, everywhere, to learn the translational approach, yeah. We all eat bananas, the banana is hanged from the top with the tree and we just take the harder part, we remove it. But have you ever seen how monkeys, they eat banana, they eat other way around. The soft part they smash it and peel it like this, and the handle is used for eating banana, yeah. So, monkeys, they know better how to eat bananas, you know, because when we eat it at the end, it always slips out. When you use it other way around, you just observe monkey how they eat banana. That is a better way of eating banana, they know it, yeah. Have you seen the banana, right? You have a banana which is there in the center stalk. We always try to break it from the stalk and you just crush bit of banana. Whereas if you reverse it, the softer portion, if you just smash it slowly, and then if you remove it and eat it, and you have full handle in your hand and you won't drop it, and then it's easy to throw the peel as well, yeah. So it's not that only humans teach animals, sometimes animal teaches us as well how to make use of it. There is a story in Africa, when there is no water, they intentionally feed some of the monkeys, the baboons and certain animals with the salt and all, and then they observe that particular animal, because their animal are well trained to find the water source. Yeah, you know, that's a lot of you can see lot of different types of video, and then animal goes in to get that particular water. Yeah, so they have different instinct compared to humans. Now coming back to the main point, all those things are there, technology and all, but have you thought of co-extraction? Most of the time you are doing extraction by using a water, deputatic solvent and other things, right? If I take a coconut oil or a sunflower oil, mix it with the shrimp shells, yeah, then use a technique or normal stirring at certain point, do you not think all the non-polar will come into the oil? Will it not? Yes. Yeah? And then remove the shells. Why extraction? We always have to think of the water and acid, put into the material, stir it, and then you get your extract. Freeze dry it or spray dry it, you get your bioactive. Yeah? Can we not do that way? Yeah? So if you take oil, and the problem with this vegetable oil is their viscosity, but the moment we heat it, its viscosity is significantly reduced. So if you take a vegetable oil, add shells to it, which contains not only shells, any of the biomass which we want to extract oil, either you add it or do the co-extraction with oil. After doing the extraction, get the oil away from the solid portion and look into the fatty acid profile of vegetable oil. You will see the addition of those bioactive into the oil. Simple method. Yeah. So what is the single most important principle of extraction? How do you extract it? What is the principle of extraction? Is one line method. Take what you want, remove what you don't want. So if you want to take something out from a bag, you take that particular stuff. To take it out, you need a specific conditions, or else remove all the bag. So always extraction is done for the targeted compound. Identify the compound, its properties, based on that you design your strategies. Yeah. So some are thermosensitive compounds, so you cannot use heat. Some are heat can resist a high amount of heat, there you can apply the temperature. Yeah, that's the way we separate the protein from oil, whether it comes to butter oil, yeah, that is ghee. And then these are extraction technique. You can get protein, peptides, collagen derivatives, chitin, chitosan, enzymes, oil, polysaccharides, biofuel, minerals, and whatever the water you have can be through a membrane processing. We will cover it again in tomorrow's lecture, and this can be used to grow microalgae, yeah, macroalgae or duckweed. Yeah. So that was the reason of showing you those microalgae slides in the beginning when we are talking about waste valorization strategies from, or quantifying waste from farm to fork. Yeah. Otherwise it becomes slightly confusing how we get the microalgae. Yeah. Because in a given short time frame we have to connect all the dots at the same time to link with different biomasses. Yeah. So this is one of the strategy which can be used for achieving a zero waste strategy in terms of recovery all the nutrients, and then same thing can go with the extraction technology. Yeah. So that's it was so neat, and I spoiled it with all the text there. Yeah. Now, what are the current valorization strategies for different stages? One is your aquaculture. Aquaculture, that is production. Either you use a pond system, what your colleague mentioned this morning, or using a recirculating aquaculture system, that is water circulated through. What is done currently, whatever side stream, dispose, yeah. That's why all the area of seafood processing is having some of the smell problem. Yeah, since there is no urban laws which says, oh, there is a smell is coming from the factory side of it. Yeah, you go in a dairy plant, you will see some paper mills. You know, oh, the smell is there because of paper mill. Yeah, so if those regulations are there, then you have to see how you dispose it properly so that it doesn't causes air pollution with those smell. So here is uneaten feed and sludge. These are the byproduct what we get during the production. Yeah, when it is in free water and free ocean, all these byproducts are consumed by some other microorganisms, and there is a self-balancing act. Yeah, uneaten feed and sludge, byproduct at the bottom of the system where microbes convert it to less toxic material. Yeah, how we can use it for growing the bacteria or microorganisms, or how you remove through sedimentation, treatment in wastewater. Yeah, so these are the management strategy, but still the problem is how we solve this aquaculture. It's not quite easy to recover that byproduct and valorize it, because these are in aquaculture system. Yeah, so that's why new strategies are coming up for production system. One of them was IMTA, because some of those byproducts can be utilized by other species. Yeah, that's why integrated multitropic aquaculture system is coming in place, and within that there is also organic certification. That means whatever is produced is organic in nature. Yeah, because of different system and no chemicals or pesticides used. Second one is fishing. Yeah, that is, one was fishing means wild harvest, no treatment, dispose, mainly bycatches, return of dead catch to the sea. Yeah, so that is still not advisable. Then again you are disturbing the ecosystem. Fish meal production. So whatever bycatches you are getting, you can make a fish meal. Applicable also for bycatches, that is production of fish meal in processing plants required for aquatic feed product. So whatever fish, small and bycatches, if you cook it to 70 to 80 degrees Celsius, yeah, whole of the fish as a biomass, either you centrifuge it or you squeeze it, you get all your oil. As what you mentioned about microwave, you can cook it using microwave, or you can cook it normal cooking process. When oil is removed, whatever is left over, if you dry it, you get a fish meal. Yeah, think if fish contains 14% protein, yeah, 70% moisture, just leave it only these two figures. Yeah, makes it at the moment how much? 84-85%. Yeah, and suppose 10% lipid, rest is ash. If this biomass is dried completely with the 2% moisture, what will be a protein content then? It will cross more than 50%. Yeah, when you extract a fish protein, if you get 60%, so simple strategy is to dry a fish, grind it, use a particle size separator. Fine particles will be all your protein, coarse particle will be bones and scales which are not grounded. Yeah, that is called dry way of extracting protein. Your colleague who is extracting protein from pulses, yeah, is a dry fractionation or a wet fractionation. In dry fractionation, there is another technique called air classification. That means you make very fine flour, and then you have air cutters which separates the starch, protein and fiber based on their densities. Yeah, that is other way of classification. So if you are drying, if you are making a meal of the whole fish meal, you get your oil out by squeezing, then oil and moisture is reduced, then you have your fish meal with good amount of protein in it. Then that particular biomass, dried biomass, can be used for production of your peptides or to obtain a purified protein. Yeah, So you design your extraction strategy based on what is intended application. Yeah. Otherwise, protein extraction from fish is very straightforward. There is not much required. You have a fish biomass, you add an alkali, within 10 minutes all the protein will be solubilized in alkali. Yeah. And then you use acid to precipitate a protein and you get 70 to 80% protein within from fish. Very straightforward procedure. Yeah. Similar process is used for alkaline protein extraction from cereals and pulses. Yeah. Anybody want to add anything to that? If you want to make it complicated, you can make it complicated. You have to increase efficiency by using ultrasonics and all. Yes, that can be done. But in order to use a technology or any process, which is important for you all to understand what is the basic process first. Yeah. I have seen lot of work is being carried out for fortification of bread with A flour, with B flour or C flour, and then you compare, we can replace wheat flour with 5% of tomato flour, banana flour, whatever you want to call it as. Yeah. But before making fortified bread, learn how to make a bread. Yeah. That is the biggest challenge. Once you know the common process, in order to extract chitosan with microwave, ultrasound or whatever technique, first do the normal extraction and then to see where you can use the technology. It's a single step extraction, that's okay. But if you have multiple stages of extraction, what you will see in the coming lectures, then you have to run a control process and to see where all you can improve the efficiency. Grinding can do the job to improve the extraction. Yeah. Same thing for extracting oil from sunflower or canola or mustard seed. You have a seed, you put it into a small bit of water. You have seen a farmer like, what do you call that, kolhu ka bell. Have you heard of this word, kolhu? Yeah. How the oil is extracted in villages and farms. Not the machine, kachi ghani, even like KTC is there in KTC in Kerala. They extract lot of oil, they export it to Europe. Yeah. How oil is extracted? You have the seeds, you press it, either hand pounding and you press it, you get your oil extracted. Yeah. But in order to improve the efficiency, you sometimes toast it. Yeah. Or grind it to improve the efficiency. And then other one are supercritical and all. So what happens? We always think at 4th level solutions, but majority of the solution can be achieved at lower level itself. Yeah. Technology is there, but to be used based on the requirement. Now coming back to this fertilizer production. So fish meal is one of the most low hanging fruit which can be produced, and same fish meal can go for pet food industry or for feed industry, including it will leftover oil is providing your essential oils and everything. Yeah. And then if you are making a fish meal from whole fish, then it can be used directly for food application. Leftover biomass and other element can go for pet food, animal feed, or for fertilizer production, or liquid or solid fertilizer, silage, that means using that biomass into acidified environment is a silage. Yeah. Or compost through fermentation. Other one is mitigation, that is prevention. The famous saying, prevention is better than cure. So how you can avoid bycatches? So you make your, if you are targeting cod for example, cod is a big fish, or you are targeting a mackerel, why you want to use a smaller mesh size? You can have a bigger mesh size. Yeah. So that small fish are not caught. So that is what it means by mesh size. Yeah. Use of fish eye devices. There are a lot of techniques are there which focuses or only captures specific type of fish and let go other fish. Yeah. Implementation of circular hook or alternative bait in long line fisheries. Only use a bait that is which attracts a specific type of fish. Yeah. Make sense? Or improvement in remote sensing and animal tracking technologies, drone and all those AI mediated systems, all these things can be. Yeah. So that means how you can avoid bycatches. If you cannot avoid it, you have to make use of it. Yeah? Does it all make sense? Is it easily said than done? Yeah? So the best part here itself is when you are harvesting it, because there is also a problem. Harvesting a fish is done by A agency and processing is done by B. So their target is only to harvest maximum what they can and hand over to the processor. Since there is no regulation about bycatches, the moment regulation comes in, whatever you are harvesting from the sea, you cannot dump it back to the sea. If you regulate it, then you will focus on these type of processes. But even whatever bycatches are coming, and if you make a simple process of making fish meal, it will add to the economic benefit of fishermen, yeah, and then they can sell that particular product in a market. For instance, if you go in some of the supermarkets, you have seen like rose mix as a fertilizer, yeah, buy it and analyze it. It's a chicken litter, yeah. Cactus powder, what is it? Broken bricks powder, yeah. So this is all our innovative way of selling it. Or if you have that same water coming on the fish side of it, contains liquid nitrogen and different minerals, that is sterilized, packed and sold as a liquid fertilizer, yeah. Now processing, 2 stages. One, don't do anything, no treatment, dispose head, bones, offal and skin, quite common practice. Here processing could be at HORAC sector, that is hospitality and catering businesses. Waste is eliminated through its discharge into ocean or is disposed in landfills, yeah. We do this maximum time, yeah. Other one is valorization, that is using a biorefining technique, get solid and liquid waste and extract high value ingredient, and this is the focus what we'll have for next couple of lectures. Other, consumption. So this is the question for you guys, how we can reduce wastage at consumption level because you are the consumers, or how we can valorize it at a consumer level. This is even more challenging, yeah. So that means you have to segregate organic, inorganic waste, but within organic you have a plant and animal. How do you do those segregations, yeah? And that could be very, very interesting project, interesting ideas, which you have some ideas you can propose. Government is currently very promising. They do promote new businesses to startups, and a lot of talent pools are there, as what I hear. And you come up with that strategy, and you'll solve not only the problem, but at the same time establish your business, yeah. And required scientific knowledge you already have. You don't have to read more books and papers to get to that one, yeah. And this is all what you see is from one of the publication which is authored by our Irish Seafood Board, that is known as Board Ishq Mara. That is not the Ishq what you see in a movie, that is the way it's an Irish word, BIM.IE, that is Irish Seafood Board, yeah. So this is all different strategies whatever is done, and you can look into that particular website which talks about different projects, different valorization strategies, and what they do with lot of fishermen, aquaculture and different farmers. So now coming back to solid side stream and liquid side stream, and you will be seeing this particular slide tomorrow again when we go for liquid and solid side stream. So this is again rough estimates how much are the solid, yeah. Overnight when you have a time, just think how we can make use of it, segregate it and get different product. Get the tangible one, not the book knowledge. We can get the chitin, we can get a collagen, but how we can get it. Again, when it comes to water, that is washing water, blood, waste brine, if you are brining the meat, cooking water, drying water and fillet. Yeah. And then if you will see all the fish waste, water, carbohydrate less than 1 because it does not contain much of carbohydrate. Whatever carbohydrate is also because of the digesta and what is there in their stomach, protein, fish, classified lean fish, that is less amount of fat, or fatty fish. How many of you have seen cod liver oil? Yeah. How it is extracted? Liver is a byproduct. Yeah. So popular, isn't it? Very expensive oil. Yeah. So same way cod liver oil is there. Why we can't have a mackerel liver oil and all, right? So basically it contains specific type of fatty acids what we briefly touched in the beginning, and all these oils can be easily segregated, fractionated to have number of different products. So this particular slide you will see tomorrow again when we go into details of side streams. Yeah. Now this is a seafood processing chain. So you have a fish, yeah, bycatch, whatever, transportation. This is normal bycatches which are obtained based on current production facilities or current mesh size, and then once it is processed, filleted, that means you remove this portion or you remove this portion. Yeah. This byproduct generation could be ranging from 20 to 70%. Yeah. That also depends upon your automation level. If it's normal automation, you are only cutting this middle portion. If fish is big, yeah, you don't have a proper grading, you have more losses or more byproducts. Yeah. But remember, the logic is even this portion of a fish, oops, let me draw the fish. How we used to draw fish? Yeah, I can make eyes as well, right? This is fish. Yeah. If you cut this portion and this portion is used for filleting purpose, composition of this portion meat or muscle will be same as here and here, right? So byproduct is having the same value in terms of protein and composition compared to rest of the material. Do you all agree with that? Yeah. Because same type of muscle, but obviously composition bit are different. And then you fillet it. Then you get fish scale, blood and sludge byproduct. And this byproduct goes as at the moment as a landfill or is disposed. And this is low hanging fruit where you can extract your different ingredients out of it, and very simple process which will try to go through with different techniques, technologies, valorization approaches. And other one is this fillet goes for different type of processing to produce products. Yeah, fish cakes, you have fish fillets, ready to eat, RTE, that is ready to eat food products. RTP, that is ready to process, and I will add new word RTC, ready to consume. Yeah, there is no point in making RTD, ready to digest. Yeah. So basically all these different product, then you increase level of processing, and during all these processes you get your commercial food product. Yeah. Commercial food product, and you have a wastewater. Yeah. Correct. And how we make use of this wastewater, either processing wastewater or solid waste. So yesterday when we are going through on the case study, I showed you a fish, different product coming out of the fish, fillet, sausages, another one, and then you have a different ingredients, and then you have a water valorization. Yeah. So how these different things are interrelated and an important aspect is when they are interrelated, so we have to always go back and come back to the new stuff. Something like you are reading a book, a lot of abbreviations are used. So, every time you go to the first page to see, oh, what does it mean, and then you go back to the lecture or go back to the chapter. Similar thing happens in this particular lecture moving forward. We will be going to and fro. I will try to calibrate all those some of the older slides into it, so that we get our refreshing, we get refreshed with what we have studied or gone through previously. Now, again, waste generated aquaculture or capture fish, bycatches, on-board storage, on-board handling. So, these are the technique which are required for reducing the wastage. Yeah? Frozen storage, chemical preservation or different stuff for capture. Yeah? Aquaculture, how best you have post-harvest handling facilities, sludge and fish mortality. That means, when you are doing aquaculture production, one of the big challenge is mortality of fish. If there is any disease in a fish, how that can be spread and how you control it. Then again it comes with your antibiotic and chemicals used. Then again we are adding another issue with aquaculture, right. So, aquaculture is better than wild harvesting, but it is having its own challenges. Yeah. And in some cases, to reduce this mortality, have you heard of lumpfish? Is also known as cullfish. These are very small tiny fish which is injected with, which is vaccinated. Each and every fish is vaccinated and is used in salmon farm to eat lices which are there on the big fish. With the mackerel, salmon, all the lices which are also known as lump suckers, to eat those lices till the time they are 100 grams, and after that they stop eating those lices, and those fish are released in the water and they grow as big as few kgs. It can go up to 10 kgs or 15 kgs, or we call it as a cull fish. When their job is done, those small fish are removed, they are, cull means killed. Yeah? But that are also can be valorized for different product. The argument of culling is because they eat lices, so they are not fit for human consumption. You can agree with this argument, you can disagree with this argument. In that case, chicken which eats all the insects should not be consumed. Yeah? So, that is the logic, because leave the chicken eats all the insects, yeah, and all this whatever chicken curry, chicken, butter chicken, all those chicken 65, all those ingredients are there, yeah. Then we should not be consuming that particular chicken, yeah, because chicken is produced by consuming insects. So, that is the name it gets from lumpfish, and this is one of the biological way of removing your lices from salmon farm, yeah. So, that is all how different storage. If you Google your cullfish or lumpfish, you can see how they are grown and where they can be used in aquaculture system. Yeah. Have you heard about this lumpfish before? It is quite prominent in England, Scotland and Ireland. Is produced and is sold. It's quite expensive, and each and every small tiny fish is hand vaccinated. And they need around more than 50,000 or 100,000 of those fish. Each and every fish is individually vaccinated, so that they do not get a disease and spread it into aquaculture farm. Yeah. There is a saying, you know, one fish spoils everything. Yeah. Same way, how do you control the fish mortality is an important one. Same thing, mortality issues are also with mussels. Yeah. As well as other things, because you can't go and check the disease, yeah, in aquaculture, because it's a standalone. You don't go there, it has to have a way of checking it, yeah. So, every new technology will have its own caveats which has to be addressed. Now, then when it comes to processing and storage, fresh fish, you are drying, smoking, salting, canning, yeah. Freezing, salting, again you have a byproduct, and wastewater. This is a rough estimate of wastewater what is generated per kg of fish. For every 100 kg of sardine, we need at least 1000 meter cube of water to be processed. So, amount of water required for each and every operation, you can quantify that water required, yeah. So, that is why one of the biggest strategy or requirement from the industry is how to reduce the utilization of water from the processing chain. That means, how we can reduce the water or reuse the water, yeah. If the water coming from here can be used for washing of equipment or to reuse the water, because water is a precious commodity, and then retail, and then 10 tons, then you lose 3 to 3.5 ton on board line processing and retail. So, it shows how much losses can occur. So, all these values are different. Need not to remember those values, just remember approximately how much losses happens, yeah. 100 kg of fish, if you have to extract a protein, maximum protein what you can get is 14% out of it, yeah, that too on a fresh basis. So, that is why material balance is important, to know what all you can obtain, then only you can make an economic value of it, then only you can propose a solution. So, what we all work on a specific unit operation, but we have to make a full process and then worry about transportation of those processes. The one what I was talking about, that BIM paper, is from Journal of Cleaner Production, 392/15/2023, and you can see exactly the same figures over there, yeah. And there is a couple of chapters and our own books are there, which also mentions pretty much similar stuff. And now, if you see high added value product, pharmaceutical, cosmetics, biotechnology, you have seen the diagram in the seaweed as well, food, animal feed, fertilizer, energy. That means, what all you can get out of it, that is collagen, chitosan, gelatin, different side streams, protein, hydroslate, oils, pigments, fish meal, that is fish meal flour, that is making a flour out of fish, fish oil, protein hydroslates or protein concentrate. So, assignment for you tomorrow, I will ask a question, what is the difference between protein isolate, protein concentrate and protein hydroslate, yeah. What is, and dairy people, they know, they should be knowing this definition very clearly, yeah. And what is a protein rich? When you say about alternative protein, fish contains only 14% protein, yeah, correct. Egg contains 4 or 5% protein, or milk contains 3 or 4% protein, whereas wheat contains 12% protein. But why egg protein is better than wheat protein, yeah. And that is a big word, yeah. Why bioavailability of egg protein is better or fish protein is better than wheat? Even if you mix wheat with dal or rice with pulses, you get all your amino acids, yeah. Very simple, you know, because we are animal and these are also animal. So, animal protein will match animal, yeah. That does not mean if you eat a human meat, it will have a better bioavailability. It is just because the configuration is the same, yeah. When we go for it's just a logic one. You agree or you disagree, either convince me or get convinced. So, when we get a plastic surgery, so we use the skin or we get what you call bypass surgery, vein is taken from your own body, is not taken from some other person's body, yeah. Same way there are plant kingdom and animal kingdom, that is the way system has been divided, yeah. So, that is why it is having a better utilization on human system, yeah. So, that is why it's, and other things are about amino acid profiles and everything, yeah. and these are readily digestible ones because they are ready. You can, even if it's raw is eaten, it's still perfectly fine, but whereas raw grains are eaten, they have to be processed in different manner. So coming back to this one, is waste from the byproduct of interest in a circular economy strategy to add value to the end product and achieve zero waste. This is very famous triangle, yeah. Waste not, want not, recycle, reuse, repair, and all those words are there, yeah. But when it comes to actual practice, it is quite challenging, yeah. So if you do it, then you will have completely different idea, yeah. Because I do know to use different enzymatic pathways, supercritical ultrasound, fermentation, microwave. What are the challenges? It's easy to do it in 100 grams, 200 grams. When you are talking about the tons, yeah, tons of fish harvested, how you are going to do that, yeah. Scale up of processes, scale up of technology is a biggest challenge, and then adopting those processes at commercial scale is another challenge, and then convincing the industry to adopt those changes plus the regulatory aspect is another challenge, yeah. So technology, we throw out lot of technology, but if you see certain sectors, they make a technology, oh let's get this centrifuge technology from Russia or cryogenic engine technology from other places. It is not in a lab, it is at the existing commercial level, yeah. That means it is an operational, everything is being tested, risk assessment and SOPs, everything is there, and then you do adopt it, yeah. So there are lot of challenges are there. So how we develop that particular process, how we make a simple enzymatic processes from fish waste to produce a 1 ton of a protein, yeah. Think of the volume, yeah. So food waste by supply chain, these are EPA figures, yeah. That's why it's very less in amount. From Irish figures, primary production losses 53K, manufacturing process 230, retail and distribution 90, restaurant food services 157, household 220, total amount of wastage, yeah. So here food loss or food waste is integrated together, yeah. So you can quantify that. If I would have quantified all the waste from each and every sector, we must be talking about the percentages, and that yields nothing apart from having an information, yeah. Think of any of the biomass, if 100 kg is processed, what are the side streams they are obtained using that famous circular diagram, what is the quantity and what is the best way to extract first, yeah, and then to go to the next level, yeah. That means systematic extraction, that is known as bio refining, that is refining step by step, yeah. So again, one was reduce and to transform, yeah. So that means using better what we already use, unlocking the potential of seas and ocean, that is using well what we don't use it yet, yeah. At least after 10 days, at least this figure will be very well clear to you all, yeah. At least there will be some take home message. So this particular diagram, and to see what is already existing, how we make a best one, learning from those mistakes, whatever new biomass is, how we develop process so that we don't have to alter it, yeah. It's something like when road is built by A agency, then a telephone company comes in, digs the road and put another pipe, and then there is a patchwork there, yeah. But if you would have done in a first stage planning of the city, yeah, then we don't need to do that, yeah, or else to rebuild a drainage system. That's why there are certain cities are called planned cities. Can you name a planned city? Chandigarh, yeah. There are different sectors. University campuses, they are very well structured. You know what, where is the house, or where is the water drainage, what is the wastewater treatment, everything is very well planned. Or you have a piece of land, you just make some houses randomly and then try to connect the distribution system. So, this is what using better what we already use, or to build a new city, that is for new biomass, how we valorize every fraction of it. Yeah. Now, what could be the strategies for waste at consumer end? That is prevention or minimizing. Yeah. Embedding low food loss or waste practices, sustainable intensification, disrupting the food production dynamics. That means whatever is current dynamics is to try to change the practices to reduce the wastage. So, in order to suggest any waste reduction strategy at production or at processing, you have to go and visit the site and see what is done, and then think and come up with the solution, propose it to the industry person, then say, no, we are not doing this because of A, B and C. Yeah? When something is being done or is not being adopted because of some new reason, if you get a new idea, there are only two possibilities. One, nobody thought about it, or somebody thought about it and it didn't work. Yeah? So, if you do discuss that idea and then you say, no, we did try but it didn't work because of A, B or C, but you may have a solution for A, B or C. Yeah? So, that is the only way what it means objective. Goals, adoption of low food loss or waste practices, infrastructure as you mentioned about the cold chain and all, that is done at larger scale. Stakeholder collaboration, that is stakeholder, that means all the people who are involved in that particular one. Trade through short value chain, if the producer is a processor, yeah, so you are eliminating the distribution and losses. Increase the capacity for food processing, that is why a lot of food parks and everything is getting established, different locations, and sharing of support for food loss and waste. That means, if a company is able to reduce it, how those practices can be shared. So, there comes the trade and intellectual property rights, yeah, so that those informations are freely available so that other peoples can adopt it. Because of that, industry, academia collaboration, international projects happens to have to share the best practices. Yeah. And then policy actions, that means you design different policies, and this is the area which I am not so happy with to go in, because making a policy is easy, implementation of those policies is difficult. Can you name a policy which is very easy to say and very difficult to implement? Honesty is the best policy. Yeah, this is what we have been hearing right from the childhood. It is very difficult to implement it. Yeah. So, then going back to the same waste hierarchy, most preferred option is to prevent the food losses and waste. Whatever is there, to reuse it for human consumption. What cannot be used for human consumption is use it for animal feed. Yeah. But nowadays feed regulations are much more stricter than food regulations. Pet food industry, pet food is more expensive than in some cases human food. Yeah. And pet food sector was always considered as recession free business, because people, they can reduce their consumption, but they will definitely feed their pets, and with that too with high quality feeding material. I can give you numerous examples. Couple of companies from America were with us. They wanted to use certain technology to improve feed quality. And if you do the microbial assessment, quality assessment of animal feed, what is sold in a market and a food, you will see they are much more microbiologically stable compared to human food. Yeah. Recycle and reuse. Recycle for nutrient recovery, this is what we will go through for microalgae cultivation, macroalgae cultivation or duckweed cultivation. Leftover recover energy in a form of anaerobic digester, biogas, methane production, and then biofertilizer. Last one is to dispose. Yeah. So, this is again European Commission Joint Research Center, that is again is a European wide policy, and you would have seen even the similar type of diagrams in some of the multinational companies what is their waste charter or waste reduction strategies. Yeah, if you go to the hotel, it says we do not clean your room every day or towels every day. If you want to get it clean, put it at one side. Yeah, just because they wanted to reduce the usage of detergents and water. Yeah, so different sustainability agendas are there. Now, same thing when it comes to fish. You have high value product, low value product. Its triangle is inversed. Yeah, what other is the difference here? What is the difference between this and this? This is general purpose. Yeah, when it comes to meat and fish, there is another issue of category, that is high risk product, medium risk or low risk. Yeah, you would have heard mad cow disease. Yeah, right. So, when we are dealing with muscle food or high risk food product, and these are the one which can carry the foodborne pathogens, botulinum and all those things, and which is very, very high risk, and it has to be disposed. Yeah, that means it has to be incinerated. If there is a dead animal, it cannot be valorized. Even though if you are valorizing it, it has to be incinerated. All this COVID virus, we all know all those stories. Yeah, so that's why it becomes quite tricky when it comes to different categories of waste. Yeah. So, and it is not that you have high risk, you have to dispose only, but still there can be a valorization strategy related to that. So, all this remains the same. Yeah. The only difference between the previous one and this one, this is upside down. Yeah. If that was M, Mohan's mother, and then it becomes Mohan's wife. Yeah. Triangle is upside down. The only difference is this is related to high risk meat products or muscle food. Yeah. Oh, sorry. So, when it comes to fish loss and waste, primary production, value chains, different activities related to that. Post production, landing, handling, storage and transportation. Processing, cutting, drying, fermentation, salting, different steps. Yeah. Distribution, retail and trade, and consumption, storage, preparation and table. That means that is only way how we can reduce our wastage. Primary, post production, causes of food loss and waste, and I will give you the reference of this particular book chapter which you can read it, and this is written by us and our colleagues. And processing and cause of losses, distribution, loss and waste, and consumption. So, it is pretty much the same thing in a different formats. Yeah. So, what is the take home message? How we reduce wastage. Yeah. So, tomorrow will or what we have covered so far, all different types of waste, side streams, whatever is obtained, and how we can create a value chain. As you will see, lecture number 17, 18 are all related to value chain creation coming from the side stream or the product made out of those ingredients, and how those ingredients can be extracted, and how we can come up with integrated biorefinery system. That will be covering from tomorrow for solid waste, liquid waste, and production of biomasses, that is duckweed production, microalgae production, macroalgae production. Yeah. So, some of the boring stuff is already been covered. So, it will be something what you will see. Yeah. And what we have covered so far, how do we first quantify the waste along the chain, that is very important. Yeah. Quantification and classification of waste. Yeah. That means whatever waste is generated along the chain, you need to classify what are the type of waste, category of that waste, if having high risk or low risk, and what can be used, and then how that particular stream can be converted into different ingredients and food and feed purposes, yeah, and we'll go into depth of this tomorrow onwards, yeah. Any questions? No? Close it then.